So good morning everyone. I am Dr. Chithra Menon, your Medico faculty. So today we'll have a rapid revision of all patho topics. So first of all, I want to tell you about the aim of this session. The aim of this session is to give you an eagle eye view of everything that has something to deal with pathology. I will not be going into depth into each question, but we'll just gloss through all the questions and I will tell you which all points you need to focus on. So coming to patho questions, you will have three types of questions in pathology. The first one is going to be your conceptual type that I hope to go through all these concepts again today. The second one is your knowledge type. And the third one, knowledge type, is those are the kind of questions that you need to memorize, and those you'll have to make them tables and you'll have to go through it either on the day before the exam or on the day of the exam. I will also be dealing with that. And the third type of questions is your integrated questions, wherein you'll have questions which will either from your medicine or your surgery or your pediatrics or your gynae, where you'll get some clue from the question which is a clinical type and some pathological clue will be there which will lead you to the answer. Okay, so without rambling on, we'll move on to the first question. So, which of the following is the characteristic of irreversible injury on electron microscopy? So, before we begin, you have a cell. You have a normal cell in which the normal homeostasis is taking place. Suppose this cell undergoes some sort of stress. Either it can adapt or it can go into some sort of injury. If the stress, that is, if it is a noxious, very potent stress, it can directly go into injury. Otherwise, if there is a stress, the cell can try to adapt, and if the adaptation is not working properly, again it will go into cell injury. Now your cell injury, you will have two types. The first one, you will have a reversible cell injury. So that is the cell, the body is going to make sure some factors, some cells are all going to come and they are trying to control that injury. If the body is successful in that, it is called reversible cell injury and the cell will move back to your normal homeostasis. Suppose the noxious stimuli is so big that the body cannot prevent or cannot do anything for the cell. What is going to happen is your irreversible cell injury. What do you mean by this irreversible cell injury? Essentially, it means cell death. Okay, cell death. Now, this cell death, it can be of various types. You can have necrosis, you can have apoptosis, then you can have something called necroptosis, then you can have pyroptosis, then you can have ferroptosis. All of these are types of cell death. We'll be dealing with each in detail later. Now going on to this part, the adaptations. How can the cell adapt? The cell can increase in number. That is called your hyperplasia. The cell can increase in size. That is called your hypertrophy. Then the cell one type of cell can change into another. That is, one cell changes into another. That process is called your metaplasia. And then you have another process in which the cell becomes shrunken, it shrinks. That is called your atrophy. Okay. All of these can occur in various physiological and pathological conditions. So this is in short what is happening in the cell. Now, here, what are the causes of your cell injury? So just imagine, what are the various injurious agents you can think of? You can have hypoxia, that is your oxygen deprivation. Then you can have some physical agents. What are some physical agents? You can have something like heat, then you can have mechanical trauma, and then you can have radiation, anything like that. Then you can have some chemical agents. Your chemical agents, the most common one is your poisons. That is, you can have your cyanide, then you can have some cytotoxic cell poisons, other things like that. Then it can be because of infections. So infections, what are all the different kinds of infections? You can have your bacterial infections, your viral infections, your fungal infections, your parasitic infections, and your prion infection. And then the cell can have some genetic abnormality that we'll deal with later. And then it can even be nutritional. Okay. So all of these can cause a cell injury, and what the cell is going to do after that injury is what this particular topic is all about. Now, if you take a cell, another thing I want to tell you before I actually forgot to tell you is that I want you to write along with me as much as you can, because I would rather you listen and write rather than you listen passively. And second thing is, all the figures and all the diagrams that I have included in this PDF, which is linked in the description, is from your Robbins, and that is the standard textbook for pathology, and that is the one resource that NEET-PG says is where the questions are from. Reference for NEET-PG, pathology reference for NEET-PG is your Robbins, and all the figures and all the diagrams that I've taken are mostly from Robbins. So if you are able to go through it once before you go into your exam, it will help you. Okay, so this injury is time dependent, and because injury is taking place in the cell, which is at the microscopic level, you can have several types of changes that are taking place in the cell over a period of time. So if you draw a graph like this, you write your time, that is the duration of cell injury, whatever it may be, and if you write this is your effect. So first think, cell has a function. So if there is an injury taking place, what do you think is going to happen? The cell function is going to deteriorate over time, right? So you have a deterioration of the cell function over time. So this is your cell function and it is deteriorating. Now, the first change that is going to take place inside the cell is going to be a biochemical changes. It is not going to be visible to the naked eye. So, first you have your biochemical changes that is taking place inside the cell, which is not easily visible to the eye. And then you have your ultrastructural changes. What do you mean by ultrastructural changes? Consider a cell. You can see the cell with the naked eye. You can see something with your naked eye, then you can say it is visible to the naked eye. Now, if it is microscopic, there are several types of microscopes that we can use. If a simple microscope is able to pick up the changes, it is called light microscopic changes. If you need a magnification higher than the light microscopy, that is, if you need something like your electron microscopy, that is called ultrastructural changes. So imagine, in the cell, the first thing that is going to take place is your biochemical changes. And after that, you will have your ultrastructural changes. Again, you are not able to see it through your light microscopy. You need something bigger than the light microscope. That is, you need something known as an electron microscope. So that is your ultrastructural changes. And after that, you can have something where you can have your light microscopy. That is the normal microscopes that we use. It can pick it up. That is your third type of changes that is going to happen. And finally, you will have something that you can see with the visible eye. That is your gross changes. Okay, I hope that is clear. So moving on, we are talking about injury, cell injury, which is of two types. Reversible cell injury, in which the cell reverts back to its normal homeostatic position, and you have irreversible cell injury, wherein the cell undergoes death, which is by various types like necrosis, apoptosis, necroptosis, etc. So your reversible cell injury, you can have two types of changes. It can take place in the same cell or it can take place in different kind of cell. The first and foremost and the most common and the most important change in a cell that takes place is a generalized swelling of the cell. Okay? And why is there this generalized swelling? This is your cell. Inside your cell, you have something known as the sodium-potassium pump. The first change that is going to take place is the sodium-potassium pump is going to stop working. It has stopped working. So it is because of this, it is because this sodium-potassium pump is not working. That is the reason why you have your swelling. Okay. Then consider a cell. Now this cell has swollen in size. When suppose you take a balloon, you fill it up with water. What is going to happen? That balloon is going to have a turgor, right? Turgor is anything where when there is some liquid inside some structure, that liquid is going to give some pressure to the structure that is surrounding it. That is what is known as your turgor. So you have a cell that is swollen, then you have something called increased turgor, and then you have an increase in weight of the cell, and it is going to appear pale. All because of this generalized water, that is this fluids that has come inside the cell. So that is the first type of reversible change. Now suppose you have some cells which has some fat molecules in it. So it is this fat molecules that is going to respond first to the to the noxious stimuli, whatever it may be. So that you can see as fatty change that is taking place. Now the very beautiful term that we use for fatty change is steatosis. Steatosis. And this is usually seen in your hepatocytes, then your beta type of pancreatic cells. Okay. So, 1 minute, let me just add a page. So, where we have reached right now is, you have cell injury, wherein you have a cell that is swollen, which is the first type of injury that is there. Then you have an increased turgor because of the water that is present, the fluid that is present inside the cell is going to apply pressure on the cell membrane, that is going to increase your pressure, that is increased turgor. Then you have something called increase in the weight of the cells, and it will be pale because of the extra fluid that is there. In microscopy, what is going to happen? Okay, so in microscopy, as I said earlier, you can have two types of microscope. Light microscopy is your usual microscope that you see everywhere, used in labs with two binocular vision and all that. And you have electron microscopy, which you use for ultrastructural changes. So in your light microscopy, this cell swelling and all, it is going to be visible as a hydropic change, which can also be called vacuolar degeneration. What is meant by that? Here you have a cell which has undergone some injury. You will have some vacuoles that are there inside the cell. What are these vacuoles composed of? This is your nucleus and these are your vacuoles inside the cell. These vacuoles are composed of the endoplasmic reticulum that has broken off because of this injury and the things that are taking place in the cell because of this injury. Okay, that is the first change. That is, on microscopy, you can see vacuolation. The second thing is, the cytoplasm of the cell becomes more eosinophilic. So consider a cell. you have a cytoplasm and you have a nucleus. In normal HND, that is in your normal staining, which is hematoxylin and eosin, your cytoplasm is going to be pink. That is, it is going to be eosinophilic. And your nucleus, it is going to be a dark purple-like color, that violet color. And that is called your basophilic nucleus. So, because of the fact that in the cytoplasm, the cell organelles are undergoing stress and are breaking down, there will be decrease in RNA inside the cell, and this RNA is a nucleic acid, and that will impart some sort of bluishness to the cytoplasm. But because of this, because of the injury, the RNA is not working. So the cell will become more eosinophilic. So in light microscopy, two signs of reversible injury you can see. The first one is the cell is going to swell up. The second one is you can see something known as vacuolation. And the third one is you can see more eosinophilic cytoplasm as a result of loss of RNA. Okay. Now coming to ultrastructural changes. That is inside your, like when you look through an electron microscope, what is going to happen? First point, you have a plasma membrane that is surrounding the cell. This plasma membrane is going to form what is called blebs. Okay, so as a result, you have something called blebs that are formed. Then the cell margins are going to be blunted. If a cell has some this villi or something like that, all of that is going to be blunted. So you have blebs formation, then you have blunting of the plasma membrane and loss of microvilli. Okay, then in mitochondria, what are the changes that you can expect? In mitochondria, you can have some small amorphous deposits. Okay, and then you can have, as I said earlier, the vacuoles that you see in light microscopy. These are the parts of NER, that is the endoplasmic reticulum, that is broken off. So you can have dilatation of the endoplasmic reticulum, and you can have detachment of the polysomes that are attached onto the plasma membrane. Then you can have some sort of nuclear alteration. That is, as I said earlier, you have a cell inside which you have the nucleus. Inside the nucleus, you have very tightly bound DNA material. Now, because of cell injury, this DNA material that is very tightly bound, that is giving rise to your dark purple basophilic appearance, is going to disintegrate. And that basophilic stain of the nuclei is going to reduce in size, reduce in color, that is, it is going to fade. So you have nuclear disintegration. And then you have formation of something known as myelin figures. Now, what are these myelin figures? So myelin figures is phospholipids that can either be from the plasma membrane or from the cell organelle. Usually there is a misconception that when myelin figures are present, it is usually a sign of irreversible cell injury. It need not always be the case. Myelin figures as such is not a measure of either reversible or irreversible cell injury. The presence of myelin figures just indicates that the cell has undergone some sort of stress. Okay, now we'll come to the question. Which of the following is characteristic of irreversible injury on electron microscopy? Now, cell swelling can take place in reversible injury. It becomes irreversible only when the cell swelling does not come back to its original position. Swelling of endoplasmic reticulum, yes, I said there is swelling of endoplasmic reticulum, and that is what is giving rise to the formation of vacuoles inside the cytoplasm. Now, disruption of ribosomes. I said earlier you can have dilatation of endoplasmic reticulum, and you have these ribosomes that are present on the endoplasmic reticulum, and that is going to break down, and that will lead to disruption of this endoplasmic reticulum. That can also take place in both. But this amorphous densities in mitochondria is an ultra-structural change that usually means the cell is going for irreversible injury. Now, can you see this in reversible injury? Yes, but out of the 4 options, this is the most suitable answer for this question, because once you see amorphous injury, amorphous densities in the mitochondria, there is some sort of very potent stress that has taken place. So there is a chance that cell is going for cell death or some sort of irreversible injury. I hope that is clear. This is what this figure I've taken from Robbins, and this is what I have explained earlier. Then this is again a figure from Robbins, which is going to say, which is depicting the pathogenesis of a disease. Now, why I put this here is because I just want you to give a look at this diagram. Okay. Etiology, this denotes the progress of a disease. So first, you have something called etiology, which may be, etiology means the causes. It may be several causes. You can have infections, you can have hypoxia, you can have some noxious stimuli, you can have physical or chemical carcinogens or anything. And that will lead to a process by which some sort of by which the cell is acclimatizing itself to the injury and is trying to stop the injury from causing much damage, and that is called the pathogenesis. And because of this pathogenesis, because the cell is undergoing so many changes that is trying to stop the injury, you will have something that can, because of the cell changes that is happening place, this will result in changes in the normal biochemical, the molecular and the hematological parameters in a person. That is what you can see through your lab values. And after that, you have your disease. This is just, again, I just took this figure because it is important and it is there in the Robbins. First you have etiology, that is the cause. Then you have pathology, that is the mechanism. And then you have the abnormality, and that is all going to show the clinical manifestations. Now this diagram is another one that I have already explained. So you have cell function that is decreased, and then first you have the ultrastructural changes, then you have the light microscopy changes, then you have the gross morphology changes, and before that you have something called the biochemical changes. Okay, now this you have a very beautiful diagram that is representing everything that we have talked about earlier. You have the normal cell. Because of reversible injury, you have swelling of endoplasmic reticulum and mitochondria, and you have something called myelin figures. As I said earlier, it is a very common misconception that myelin figures is equal to reversible injury. That is not true. Myelin figures can be present in both reversible and irreversible injury. Now, if there is progressive injury, there is going to be breakdown of everything, and that is going to cause amorphous densities in the mitochondria, and that will lead to cell death. This is just one type of cell death, that is necrosis, which we'll deal with later. And here you have the H&E pictures. So the first one is you have a normal cell. Here you can see you have normal beautiful cells. But here in the second and third figures, one thing you can see is this plasma membrane, the cytoplasm, everything appears to be broken down one by one, right? And after that, this is your picture of necrosis, where everything has broken down. You just have the cell outline and not the cell outline in some cases. You have cell fragments there. Okay. Now we move on to the second question. I hope cell injury part is clear. Now we move on to the next question. Forensic autopsy of a 40-year-old male was done and confirmed that murder was done by smothering. If we plot the sequence of changes that occurred in the body and onset of process till the death of cells, what would be the first event? Okay. So here it is smothering. What is smothering? Smothering is when there is some pillow or some sort of obstruction that is taking place, and wherein both the mouth and the nose of the person is being covered so that oxygen inflow is not taking place. So why I chose this question is because I want to focus on two things. First one is ischemia and the second one is hypoxia. Let me add a page again. So what is the difference between ischemia and hypoxia? Can you use both these words interchangeably? No. So hypoxia simply means that there is some reduction in oxygen supply to the cell. Hypoxia does not mean that there is no oxygen supply to the cell. There is some reduction in the oxygen supply to the cell. Here, the blood flow will be maintained. So, if aerobic glycolysis is not taking place, anaerobic glycolysis can take place because blood flow is maintained. Now, ischemia is usually a result of loss of blood supply. It's a result of loss of blood supply. So, here you can both the aerobic and the anaerobic parts is not going to take place. So, as I said earlier, in ischemia, both the anaerobic and the aerobic part is not aerobic type of respiration is not going to take place, while in hypoxia, because the blood supply is maintained, even if the aerobic part is not going to take place, the anaerobic part will take place. Okay, so here you have a figure again taken from your Robbins, which beautifully explains in case of ischemia what is going to happen. In mitochondria, there is decrease in oxidative phosphorylation, decrease in ATP, and first you have anaerobic glycolysis, but once all the everything, because blood flow is not there, the products of this anaerobic glycolysis that is taking place cannot be washed off. So it will stop after some time, but the byproducts will remain. That is, your glycogen store and everything is going to stop, but your lactic acid, which is the byproduct of anaerobic oxidation, anaerobic glycolysis is going to remain in the cell, and that is going to increase the pH of the cell, and that can itself cause cell injury. Now here, because of decrease in ATP, your aerobic glycolysis is not going to take place. That is, there is decrease in the action of sodium and potassium pump. So because of the decrease in action of the sodium potassium pump, what is going to happen? You will have influx of calcium and influx of water, and you will have efflux of potassium. That is going to result in your swelling, then your loss of microvilli, and this blebs formation. Okay, again I am repeating, as a part of ischemia there is no blood flow. So you are coming to the aerobic part of glycolysis. The sodium potassium pump is not going to function. As a result of that, you can have increased sodium, increased calcium, and increased water. Also, you will have a decrease in potassium. You will have a decrease in potassium as a result of increased efflux of potassium. And as a result of all of that, what you're going to get is, you will have cellular swelling, you will have endoplasmic swelling, and you will have breakdown of the same. Now coming to the anaerobic part. The anaerobic part, what is going to take place is, you will have decreased ATP. So first the cell will do anaerobic glycolysis, then the cell is going to get energy from the glycogen that is already stored. So whatever glycogen is there, the store is going to decrease. Then because there is no blood flow to the area that is affected by ischemia, what is going to take place is, the byproduct of anaerobic respiration, that is the lactic acid, is going to remain there, and as a result of increase in acid, the pH, that is the acidity of the cell, is going to increase, and that itself is going to cause cell injury. Okay, and the third thing is, because of this decreased of oxidative phosphorylation, you will have a decrease in protein synthesis. So this is what is happening in cell injury when you look at it through the eyes of mitochondria and your endoplasmic reticulum. So again, what would be the first event? The first event is always cessation of oxidative phosphorylation. It is only after that this swelling, then this chromatin change, then this mitochondrial amorphous density and all is going to happen. I hope that is clear. Now, 2 points I have to say, 2 points that I have to tell you here is, the cell that is most prone to hypoxia is your neuron. That is expected, right? If your GIT is not going to get oxygen for some time, nothing is going to happen. But if your brain does not get oxygen, something is definitely going to happen. So the cell which is most sensitive to hypoxia is your neuron. The second thing I have to say is, there is something called HIF1, that is hypoxia inducible factor. Now this is actually making sure that this is a protein that has recently been found in some cells, which is protecting some sort of cells against this hypoxia. Now this can have future applications, because if some drugs can be there which can protect, which can increase the protection of cells against hypoxia, it can be used in therapeutics. So HIF, hypoxia inducible factor, that's all you need to know. Now coming on to the third question, true about apoptosis are all except. Okay, so first we learnt about irreversible cell injury. As I said earlier, irreversible cell injury means cell death, and it can be by various modes. It can be by necrosis, it can be by apoptosis, it can be by something called necroptosis, then it can be by pyroptosis, then it can be by ferroptosis. Okay, so we'll see apoptosis now. So what is apoptosis? From the beginning we have learned apoptosis refers to programmed cell death. So here what is happening? So you have conditions in which in the body, if there is a proliferation of cells, that proliferation has to be stopped. Now consider for example, consider your intestinal lining. It is said that over 1 million cells are being turned over every second inside the human body. So for that turnover to happen, whatever cells that is there that has lost its function has to be killed. Only then the new cells can come up and replace those cells. And one of the ways through which our body ensures that this is taking place is called apoptosis. So essentially, you consider a cell. Once the cell has done its function, once the cell has done everything it has to do, the cell is deciding it is time for me to go. That is, it is a cell-regulated suicide program essentially. Now, apoptosis can occur in both physiological conditions and in your pathological conditions. Apoptosis is not a bad thing. It is because of apoptosis that this normal homeostasis in the body is taking place. So consider what are the physiological conditions in which apoptosis can take place. First, you have something known as supranumerary cells. So what are supranumerary cells? So in embryogenesis, there is a very big type of cell proliferation, a grand cell proliferation that is taking place, but not all of these cells are necessary for the proper morphogenesis of the human body. So some of these cells which are extra in number has to be taken out, and that is one of the conditions in which apoptosis plays a role. Now second one, you take something hormone dependent tissue. So suppose you take the human breast. When lactation has to occur, the breast cells will increase. But once the baby is weaned off, all the hormones that are responsible for the lactation and that in turn is responsible for the increased number of cells in the breast will have to be killed off. All those cells have to be killed off, and that is another condition in which apoptosis takes place. Can you think of other conditions in which hormonal dependent apoptosis takes place? For another thing is, consider your ovary. Now ovary, after menopause, ovarian follicles have to undergo atresia. Atresia is essentially loss of function and shrinkage of cell. So this ovary undergoing atresia is also an example of hormone dependent apoptosis. Then another example is your endometrium during your menstrual cycle. The endometrium has to proliferate every month in order to accommodate a fertilized ovum, and if the ovum is not fertilized, all these proliferation and all these extra cells that are there will have to be shed off. That is also through apoptosis. Then you have third condition is you have cells and structures in which constant turnover is taking place. As I said earlier, consider the GIT, where you have intestinal cells and these are always undergoing turnover. Another symbol for the same is you have your thymus and your lymph nodes. Here also you have some B lymphocytes that are coming, some B lymphocytes get activated and everything happens, and if those B lymphocytes are no longer needed, apoptosis happens. Then you have apoptosis in case where there is some increased activity against the human cells. That is in autoimmune disease, not when if cells are reacting against its own cells, you need apoptosis to make sure that those cells that react against our own cells are killed off. Then after infection, so you have an infection, that is taking place in the body. As a result of the infection, you have so many cells, that is you have acute inflammation where you get your neutrophils, your monocytes, your macrophages and all those cells. And once the infectious stimuli has been outpowered, once the infection has subsided, all these extra cells that is there in the body, all these circulating neutrophils, these monocytes, these macrophages and everything else has to be broken down. That is also through apoptosis. Now those are the conditions in which there is physiological apoptosis. Now coming to pathological apoptosis, again you will have one condition in which, suppose DNA damage has taken place. Suppose DNA damage has taken place. All those cells with DNA damage will have to undergo apoptosis. That is one pathological condition in which apoptosis has to take place. Then suppose there is something called misfolded protein or something of the sort. Again, those cells have to be killed off. You have apoptosis there. Then suppose there is an obstruction to ducts. Then this is a gland with a duct. Suppose there is some sort of obstruction that is taking place. The part that is supplied by the duct undergoes apoptosis because it is no longer required. Okay. Now that is all about conditions in which apoptosis takes place. And then you have your mechanisms by which apoptosis takes place. Now, before we move on to the mechanism, we'll see what are the cell changes that are taking place in case of apoptosis. So, in reversible cell injury, we said the first change that is going to take place is your cell swelling. Here, the cell is not going to swell. The cell is going to shrink. So, the first sign is cell shrinkage. And after that, you have your chromatin condensation. Now, the word chromatin condensation, that is the most specific factor of apoptosis. So when you get a question asking which is the most specific factor for apoptosis, it is this chromatin condensation that is the answer. Okay, then you have formation of blebs. So, blebs, and apoptotic bodies. So essentially what is taking place is, here you have a cell. Initially it is going to shrink in size. Then it is going to get fragmented into several pieces. And all these fragmented pieces are called your apoptotic bodies. And in microscopy, you can see these apoptotic bodies as some granular pink colored material, that is granular eosinophilic materials. I hope that's clear. Now again, you have something called a phagolysosome formation. That is essentially a structure in which the cell is undergoing breakdown. Now, we will, one thing I have to tell you is, once all these fragments and everything that is, once the cell has become fragments, it is easily fragmented and taken up by the macrophages or whatever cells that is there, that whose function is phagocytosis. So, what is important here is that because these cell fragments do not stay in that body for a long time, because they are easily taken up, there is no inflammation. Because these cell fragments don't stay in the body to cause that inflammatory response. So another hallmark of apoptosis is that there is no inflammation. Now let's look at the process in which apoptosis takes place. Now apoptosis is mediated by activation of enzymes called caspases. Caspases. Okay. Now, one thing about caspases is there is a cysteine residue in its active site, and it is an enzyme. It cleaves a molecule, and this cleavage of that protein occurs after aspartate, like after you have a genetic code in which you have aspartic acid, you have cysteine. Now cysteine residue in caspases, cysteine residue is present in its active site and it cleaves after aspartic residue. That both of these are very different. So you have a caspase which has cysteine residue, and where this is this enzyme is going to act is, it is going to act after an aspartic acid residue. I hope that's clear. Now this cleavage, apoptosis, can have an initiation phase and an execution phase. Now, this initiation phase can be of two types. It can either be intrinsic or mitochondrial, or it can be extrinsic or death receptor ligand related. Again, apoptosis, it is by the function of caspases. You can have these caspases are enzymes which has a cysteine in its active site, and when these enzymes, these caspases, they always cleave protein after that aspartic acid residue. So apoptosis has two phases. One, you have the initiation phase, and the second thing is you have the execution phase. Now this initiation phase can be by two different mechanisms. You can have an intrinsic initiation phase, that is the mitochondrial initiation phase, or you can have an extrinsic initiator phase, that is the death ligand receptor mediated. Now, we'll take a look at the intrinsic pathway first. This is actually the pathway in which most pathological and physiological apoptosis takes place. Now, the starting point is increased permeability of the mitochondrial membrane, and it is done by some proteins, that is the BCL2 family of proteins. So again, you have a starting point, and that starting point is the increase in permeability of the mitochondrial membrane, and apoptosis in the intrinsic pathway of apoptosis is mediated by the BCL2 group of proteins. Now this BCL2 is a family of proteins which and of that family there are three different types of protein that play a role in apoptosis. You have some protective proteins, that is you have the anti-apoptotic proteins. Now here you have three. You have BCL2, BCLX, and you have MCL1. Now how to remember? It all has that C. That is, there is BCL and then MCL. It is situated in the outer membrane. That you don't need to worry about. You have anti-apoptotic protein, that is the first type of protein. And secondly, you have something known as pro-apoptosis protein. This is very important, and only two proteins. You have your BAX and your BAC. Again, BA, pro-apoptotic protein. Then you have your third type, that is your regulated apoptotic initiators. Here you have some proteins called your BAM, your BIM, your BID, then something called PUMA and something called NOXA. Again, I am reiterating. Your apoptosis, the intrinsic pathway, starting point is the increase in permeability of the mitochondrial membrane. Apoptosis, mitochondrial apoptotic pathway, is mediated by BCL2 family of proteins. Now, BCL2 family, you have three different kinds of proteins from that family. The first one, you have anti-apoptotic proteins, that is BCL2, BCLX and MCL1. You remember, anti-apoptotic is always going to have a C. Okay. Then second type, you have pro-apoptotic protein. That is again of two varieties. That is, you have the BAX protein and the BAC protein. That is BAX and the BAK proteins. Then the third one is your regulated apoptotic initiators. There you have your BIM, your BID, your BIM, your BID, your BAM, BAM, BIM, BID, then you have your PUMA and your NOXA. That is it about the proteins. Now we'll move on to the actual process as such. So here, inside this mitochondria, you have a protein called cytochrome C. Now your cytochrome C is a double-edged sword. So there you can have cytochrome C can either help in ATP production, or when the cytochrome C is released into the cell, that can cause cell death. So in mitochondrial pathway, what is happening is, this is your mitochondria. You have your cytochrome C that goes and attaches to something known as APAF1. That is your apoptosis activating factor 1. And that goes and attaches and activates your caspase 9. Now your caspase 9 is actually going to initiate the cascade of caspases, that is the executioner phase. So in mitochondrial pathway, you just need to think what is happening. You have your cytochrome C. Now cytochrome C is going to go and attach to something called APAF, that is your apoptosis activating factor 1. Now both of that is going to activate your caspase 9, and that caspase 9 is going to activate the executional caspases. Now in the cell, you have two very important proteins that are often asked as one words. You have something called SMAC and something called DIABLO proteins. Now the function of these proteins is that in physiological conditions, apoptosis is not taking place, and that is because of some factors inside the cell that is inhibiting the formation of this APAF, that is the apoptosis promoting factor. So this SMAC and this diablo protein, it will antagonize inhibit the physiological controllers of apoptosis. You don't need to know the name. Just know SMAC and diablo, it is going to make sure that apoptosis is taking place by neutralizing all the molecules that are there to make sure apoptosis does not take place. That is all about the intrinsic pathway. Now we'll move on to the extrinsic pathway. Extrinsic pathway is ligated by TNF, that is your tumor necrosis factor. So here what is happening is, you have a TNF factor receptor that is going and activating something called Fas ligand. Once this Fas ligand gets activated, you have more Fas molecules that are coming. And as a result of this Fas, you have the formation of something called FADD. That is your Fas associated death domain. Now, as a result of that, you have inactive caspase 8 or caspase 10 that is going to become active caspase 8 and 10. Now that active caspase is going to initiate the executional phase. So in short, you have the intrinsic pathway and the extrinsic pathway. In intrinsic pathway, what do you have? You have cytochrome C, then you have APAF, and that is going to bind to caspase, activate caspase 9, and after that you have this executioner phase. In extrinsic pathway, you have your TNF, that is tumor necrosis factor receptor, and you have your Fas ligand, and you have your many Fas molecules that are coming together to form something called FADD, that is going to activate caspase 8 and 10, and that is again going to result in executioner phase. Now this executioner phase, you have two type of caspases in particular. You have your caspase 6 and your caspase 3. Those are the caspases that leads to apoptosis. Now, as I said earlier, as a result of apoptosis, you have cells that are becoming fragments, and all these fragments is going to have these eat me symbols. That is, it is going to signal to the surroundings that please come and eat me. So phagocytes come and eat these type of cell fragments, and it is rapidly cleared out. Again, in short, you have apoptosis, you have BCL family that regulates apoptosis, and you also have an intrinsic and apoptosis, you have an initiation phase and an execution phase. Execution phase is same for both pathways, but initiation phase can be by two pathways. You have the mitochondrial or the intrinsic pathway, and you have the death receptor ligand pathway or the extrinsic pathway. So in the intrinsic pathway, you have your cytochrome C, you have your APAF, then caspase 9, intrinsic pathway, caspase 9. Please remember that. Extrinsic pathway, on the other hand, you have your tumor necrosis factor receptor, your Fas ligand, and that is resulting in the formation of FADD, that is Fas ligand associated death domain, and that is activating your caspase 8 and your caspase 10, and that is going to result in the activation of the execution phase. This execution phase is done by your caspase 6 and your caspase 3, and essentially what is happening is the cell gets broken down into fragments, and all these fragments is going to have some signals that is going to signal to the external environment is that you need to please eat me now because I am not good for the body. And because of that, many phagosomes come and take up these cell fragments, and as a result, these cell fragments are rapidly cleared out, which is the reason why there is no inflammation in apoptosis. Again, another thing I have to tell you here is there is something called efferocytosis, which is the process by which these apoptotic cells are phagocytized. Now what we discussed here is apoptosis, the intrinsic and the extrinsic pathway, and one word I want you to remember here is efferocytosis. That is the process by which these apoptotic cells are phagocytolized. We'll take a short break now. We'll be back in a few minutes. Okay, we'll start again. I hope everything is clear about apoptosis. I'm mentioning it once again. Apoptosis. Now, the first thing that is going to take place is you will have an increase in mitochondrial membrane permeability. Now, apoptosis is programmed cell death that is mediated by BCL2 family of proteins. In BCL2 family of proteins, you have first one pro-apoptotic, then you have anti-apoptotic, then you have regulatory things. And apoptosis, the process has two phases. It has a initiator phase and an executioner phase. Now, initiator phase is of two types. It can have intrinsic or a mitochondrial pathway, extrinsic or a death receptor ligand mediated pathway. Now, let's see the questions related to that. First question is, true about apoptosis are all except inflammation is present. I have mentioned it many times. Now, in apoptosis, all these bodies that are getting taken up easily, which is why the apoptotic bodies don't remain in the body for a long time. As a result of that, no inflammation is seen. Chromosomal breakage, clumping of chromatin, cell shrinkage. Now, cell shrinkage is there. Clumping of chromatin is the most sensitive, or you can say that is one thing which is always there in apoptosis. I hope that question is clear. Now, here you have a table. Again, all these diagrams and all these pictures are taken from Robin, so please go through them properly. You have the difference between necrosis and apoptosis. As I said earlier, cell death is of many types. You have necrosis, you have apoptosis, then you have necroptosis, then you have something called pharoptosis, then you have pyroptosis. These are the 5 types of cell death that is described in Robbins. So, the difference between necrosis and apoptosis, again, very important. Cell size is enlarged in necrosis, the cell is shrunken in apoptosis. Then, here nucleus will have pycnosis, karyorrhexis, karyolysis. We'll see each of that in detail later. Now, in apoptosis, it will have fragmentation into nucleosome-sized fragments. Then plasma membrane disruption is there in necrosis. It is intact in apoptosis. Cellular contents are intact in apoptosis, and it will go out of the cell in necrosis. And adjacent inflammation is frequent in necrosis and absent in apoptosis. So here you have the process of apoptosis. You have the normal cell, condensation of chromatin, which is very important sign. Then you have the formation of membrane bleps, and then you have cellular fragmentation. You have the formation of something called the apoptotic body, and you have phagocytosis. Here again you have the physiological and pathological conditions. We have already discussed this. Physiological condition during embryogenesis, then turnover of proliferative tissues, then you have involution of hormone dependent tissues, then you have decline of leukocytes, then you have elimination of potentially harmful self-reactive cells. In pathologic, you have DNA damage, you have accumulation of misfolded protein and infections. This is another beautiful diagram that details what we have discussed earlier. You have the intrinsic pathway, you have the extrinsic pathway. Both intrinsic and extrinsic pathway leads to caspase activation, that is the executioner phase, which is mediated by caspase 6 and caspase 3. Here you have the morphologic appearance of apoptotic cells. You have a cell fragment, and then you will have these are your apoptotic cells. In microscopy, essentially you see some granular eosinophilic materials, you have to think in terms of apoptotic bodies. Now, organelle which plays a pivotal role in apoptosis is mitochondria. Here again, difference in apoptosis and in necrosis. Leakage of mitochondrial protein, you have apoptosis. Necrosis we'll deal with later. Now this, which of the following is an anti-apoptotic gene? We have already discussed this. You have your anti-apoptotic genes, which are your BCL2, BCLX and your MCL1. And you have your pre-apoptotic genes, which are your BAX and BAK. Then you have your regulatory genes, which are your BAD, your BIK, and your PUMA and your NOXA. Here, again, in apoptosis, APAF1 is activated by release of which? APAF1, that is the intrinsic pathway, also called the mitochondrial pathway, and that is activated by your cytochrome C, which goes here. The picture is that of extrinsic pathway, okay? Extrinsic pathway is where you have your caspase, your FAD and your caspase 8 and caspase 10. In intrinsic pathway, you have your cytochrome C. In intrinsic pathway, you have your cytochrome C. Then you have your APAF1. Then you have your caspase 9. In extrinsic pathway, you have your FAS ligand. You have your tumor necrosis factor receptor. Then you have the formation of FADD, and you have your caspase 10 and caspase 8. And both of this together, in executionary phase, you have your caspase 6 and caspase 3. Okay, now this is a very important question. Ladder pattern in DNA electrophoresis. If this is your normal, this is how a normal cell looks like. A cell undergoing apoptosis will have a step ladder pattern, and a cell undergoing necrosis will have a smeared pattern. I hope that's clear. Now, morphological changes in apoptosis include, again we have discussed this several times, the answer is cytoplasmic blebs. Which of the following is the hallmark of programmed cell death? I said earlier, the starting point of apoptosis is always increased mitochondrial permeability. Okay. Which of the following induces apoptosis in a cell? The answer is glucocorticoids. Here you have a pattern, you have a very beautiful table, inducers of apoptosis and inhibitors of apoptosis. So this you just need to think logically. Now, if growth factors are present, why should the cell die? Growth factors are already present. So it is an inhibitor. Extracellular matrix, if extracellular matrix is present, there is no need of breakdown of cells to provide raw material for extracellular matrix, right? So again, apoptosis is not needed. Here, because it is inhibited, if you withdraw, it is inducing. So extracellular matrix, if you lose the matrix, there will be induction of apoptosis. If matrix is present, it is inhibition of apoptosis. Then sex steroids is going to act as inhibitors of apoptosis. That is, all these kind of sex steroids like for muscle hypertrophy and for growth and all, you need sex steroids. Glucocorticoids is going to induce apoptosis. Then viral proteins usually inhibit apoptosis, but free radicals and all induce apoptosis. Now, okay, now I hope we have learned everything about apoptosis. Let's move on to the other types of cell death. You have, now we have already dealt with apoptosis. In next you have something known as necrosis, then you have your necroptosis, you have your pyroptosis, you have your ferroptosis. So first we'll see the smaller ones and then we'll see necrosis, which has to be explained in detail. So first is necroptosis. So necroptosis, there is necro from necrosis and ptosis from your apoptosis. So it is programmed necrosis. Necroptosis refers to programmed necrosis. Now, similar to necrosis, you have loss of ATP, then you have swelling of cell, then you have the formation of reactive oxygen species. And similar to necrosis, it is also not dependent on caspases. Now, similar to apoptosis, there is a receptor and a ligand that is required for necroptosis. So here what you need to know is, you have the tumor necrosis factor receptor that goes and binds with RIPK1 and RIPK3. Now this RIPK stands for receptor interacting protein kinase 1 and 3. Now, as a result of this necroptosis is taking place, you don't need to learn about this in detail. Now, where does this necroptosis takes place? Now, in physiological conditions, it is said that necroptosis is a form of cell death in mammalian bone growth plate. Now, pathologically, you have some conditions called steatohepatitis, then acute pancreatitis, then in some cases of Parkinson's disease, then your CMV infection, then your reperfusion injuries. So all of these are places where necroptosis takes place. Again, necroptosis is programmed necrosis. Now, like necrosis, it is not dependent on any caspases. You will have loss of ATP, you will have reactive oxygen species that are formed. Now, like apoptosis, it is also a receptor-ligand interaction that is taking place, causing necroptosis. Now, you have the tumor necrosis factor receptor that is dealing with RIPK3 and RIPK1, that is receptor interacting protein kinase 3 and 1, and that is resulting in necroptosis. You have physiological conditions and you have pathological conditions in which necroptosis is taking place. So your physiological condition in mammalian bone growth plate, and in pathological conditions you have some cases of Parkinson's disease, then you have your steatohepatitis, acute pancreatitis in CMV infections. Now, the next type of thing, that is your pyroptosis. Now, where is pyroptosis taking place? Now, pyroptosis is again a type of cell death in which interleukin 1 is acting. Then you have the formation of inflammosome, and after that you have caspase activation. That is all you need to know about pyroptosis. Now, pyroptosis, the word pyro means fire. So that is, it has something to do with fever, and interleukin 1 is a cytokine that is associated with fever. So just when you hear the word pyroptosis, interleukin 1, then you have inflammasome formation, and after that you have caspase activation. Now the next type is ferroptosis. Now in ferroptosis, again you have increase in iron concentration that leads to production of reactive oxygen species, and that increased production of reactive oxygen species is actually leading to a condition in which the cell is unable to cope up with all these extra oxygen radicals that are there, and that leads to cell death. That is all. Now we'll take a look at the next question, then we'll move on to the explanation. Pyogenic infection and brain infarction are associated with, so okay, necrosis. So our next topic for the day is necrosis. Let me just add a few papers. Okay, so what is necrosis? You have heard the word necrosis thrown in all kinds of scenarios. You will say there is necrosis of tissue, there is necrosis of the wound, you have necrotic wound, you have necrotic bowel, you have necrotic tissue that has been sent for histopathology. So what is the meaning of necrosis? Now, necrosis, essentially what is happening is there is denaturation of protein. And because there is denaturation of protein, there is leakage of cellular contents, and because of that there is inflammation, and as a result there is enzymatic digestion and cell death. Okay, now this is mediated by some specific proteins that are secreted by the injured cells called DAMPs, that is damage associated molecular pattern. Okay, so in necrosis you will have some sort of denaturation of protein, as a result of which there is a leakage of cellular contents from the cell into the outside, and because of that there is an inflammatory response that is taking place, as a result of which, because of enzyme digestion, the cell gets broken down. Okay, so here in microscopy, what are the features? In microscopy, the first feature is there is going to be an increase in the eosinophilic cytoplasm. That is, the cytoplasm which is pink in color is going to become more pink in color. This is because of the fact that there is loss of RNA from the cytoplasm. This RNA is actually a substance that imparts a small bluish hue to the cytoplasm, but because there is loss of RNA, it is going to be pink alone. Then there is going to be something called glassy appearance, and then the cytoplasm is going to be vacuolated and moth-eaten. Now, these, as a result of all that, there is going to be dead cell, and these dead cells, only the remnants of the cell will be left, and those remnants you can call it as myelin figures. Now, in some cases, these myelin figures may undergo something called calcification. So again, in necrosis, there is denaturation of protein. As a result of denaturation of protein, there is leakage of cellular contents outside the cell. Because of that, there is inflammation that is taking place, and that inflammation causes enzymatic digestion and the cell gets broken down. Now, this has a clinical significance. Now, this necrosis and this increase in the leakage of intracellular proteins is actually can be used diagnostically in some conditions. For example, you have your troponins, where in case of myocardial infarction, your troponins are raised, and that is because of this increase in the intracellular proteins that are inside the cell. When the cell gets broken down, the proteins come outside the cell, and we can measure that protein, so it can serve as a laboratory diagnostic method. Now, similarly, you know, in your that is in your cardiac muscle, in your bile duct, and then in your hepatocytes, you have several enzymes called your SGOT, SGPT and all. All of these are present inside the cell because of cell gets broken down. These come outside the cell, and we can measure that. As a result, we can use that as a diagnostic tool. Okay. Now we saw what are the microscopic features. You will have an increased eosinophilic cytoplasm. You will have some glassy appearance, and then you will have cytoplasmic vacuoles. Now, in electron microscopy, what are the features? In electron microscopy, the first feature will be there is a discontinuity in plasma and organelle membranes. Then second one, you will have dilation of the mitochondria, dilatation of the mitochondria. Then you will have, again, you will have large amorphous densities. Then you will have intracytoplasmic vacuolation and myelin figures. Then you will have aggregation of these materials, that is similar to the large amorphous densities. Now, coming to nuclear changes, this is where you can have three types of nuclear changes. First is your karyolysis, then you have your pycnosis, then you have your karyorhexis. So karyolysis, again imagine you have your cell, you have your nucleus which is deeply stained blue, that is basophilic. So you have your cell that is very deep, your nucleus which is extremely deeply stained and it is clumped together. As a result of cell injury, these clumping, it opens up and it disintegrates. It disintegrates into smaller bits, and this bright basophilic color fades out because there is loss of DNA. Now once DNA is clumped together, you have a bright blue color, and when it gets lysed out, that is when it gets fragmented, you will have a loss of that blue color. So that is known as karyolysis. Lysis, that is broken down. The nucleus gets broken down and the blue color will fade, and that is because of loss of DNA. Okay, then you have your pycnosis. So in pycnosis, what is happening? Pycnosis is the type of cell injury that you usually see, nuclear change that you usually see in necrosis. It can also be present in pycnosis is the type of nuclear damage you usually see in apoptosis, but it can also be present in necrosis. And here what is happening? Here the nucleus is shrinking. So your blue color is actually going to increase because already it is clumped together and it is very blue. Because it is going to shrink, that blue color is going to intensify. So that is your pycnosis. Then your karyopyresis. Again, from this word you can understand there is some sort of breaking down that is happening. Here the pycnotic nucleus is broken down. I hope that's clear. Okay. Now we learned about what are the changes that is happening in necrosis. So what are the type of necrosis? So can you think of what are the type of necrosis? When you think of necrosis, the picture that usually comes into your mind is that of a gangrene, right? You think of gangrenous necrosis. There is no such thing as gangrenous necrosis, first and foremost. Now, what is this gangrene? Gangrene is actually your necrosis with putrefactive changes. What is putrefactive changes? Putrefactive changes means infection. That is some sort of bacteria is present. First point I want you to know is there is nothing called gangrenous necrosis. Gangrene refers to necrosis with putrefaction. Okay. Now again, when you're talking about gangrene, you will have two types of gangrene, right? You will have dry gangrene and you will have wet gangrene. Again, these are just layman's term. In pathology, these have no meaning. In layman's term, dry gangrene means you have those venous ulcers, venous stasis, that is all causing gangrene. That is your dry gangrene. In wet gangrene, you have a gangrene, that is you have a necrotic structure that has a superadded infection. Okay, so what are the types of necrosis? If it is not gangrene, what are the other type of necrosis? So necrosis, widely you can have 5 types of necrosis. The first one is you can have something called coagulative necrosis. Then you have your liquefactive necrosis. Then you have your caseous necrosis, then you have your fat necrosis, and then you have your fibrinoid necrosis. So we'll see it one by one. So in coagulative necrosis, what is happening is the cell outline is actually present. The cell outline remains, but the cellular contents are broken down. It is usually as a result of ischemia. And it occurs in your organs, in all organs. So you see a picture with any organ that is affected, and the question is any organ except the brain. That I have to say, any organ except the brain that is affected, and the question asks you what sort of necrosis it is. 9 times out of 10, the answer is going to be your coagulative necrosis. Okay, in coagulative necrosis, you have most common example is your infarction. Again, we'll be dealing with infarction later, so I'm not going more on that. So coagulative necrosis usually happens in your organs, solid organs. So the outline will be maintained. It is not that the outline is forever maintained. It will be maintained for a period of days. Only after that it will disintegrate. Now liquefactive necrosis. You hear the word liquefactive. What is the first thing that comes to your mind? Liquid of some sort. In body, what is the most common liquid of some sort that can happen in necrosis? That is your pus. So in all sorts of infections, whether it be your fungal infection, your bacterial infection, it is most likely going to be liquefactive necrosis. But there is one exception to that. Whatever is happening in the brain, that is whatever is happening in the CNS, is going to be a liquefactive necrosis. I hope that is clear. Every organ except the CNS, it is going to be coagulative necrosis, but in the CNS, it is going to be liquefative necrosis. And as the word suggests, in liquefative necrosis, the cell borders are not maintained. Everything becomes a payasam. Everything becomes viscous. It becomes like a pus. Okay. Then you have your caseous necrosis. Now, what is happening in your caseous necrosis? Caseous necrosis has been beautifully described in this condition called tuberculosis. So here, what is said is that you will have some central necrotic material that will have a cheesy, granular texture, and that cheesy, granular texture, which looks yellowish on gross section, is called caseous necrosis. I hope that's clear. Now you have your fat necrosis. Now fat necrosis occurs in conditions where the organ has fat. For example, in acute pancreatitis, and in focal areas like in the mesentery or somewhere, you will have fat necrosis. If you remember, I said necroptosis is also something that happens in your hepatocytes and all. So that is fat necrosis. Now fibrinoid necrosis happens in blood vessels. It usually has something to do with the immune complexes breaking down. Just remember, fibrinoid necrosis seen in blood vessels. Okay. So moving on to the next question, you have pyogenic infection and brain infarction are associated with, I said earlier, all types of infection and any condition in brain, it is usually liquefative necrosis. So here you have a figure that I've taken from Robbins, coagulative necrosis. I said earlier, now coagulative necrosis, usually as a result of ischemia, and that is usually called an infarct. Infarct is an area of tissue that has undergone necrosis as a result of ischemia. It can be of two types. It can be red infarct and white infarct. Now, red infarct happens in organs where there is dual blood supply. And white infarction happens in organs with end supply. That is your spleen, your kidney and everything, spleen, your kidney and all. You have white infarct, and that is coagulative necrosis. So you have coagulative necrosis here. Now moving on to the next figure, again brain, you will have liquefative necrosis, and it is going to look something like this, like some pus, some viscous material, that is liquefactive. Moving on to the next picture, you have caseous necrosis, where you have a lesion and in the middle you have this cheesy granular substance. The word caseous means cheese-like. Then you have your fibrinoid necrosis. Here you have some protein deposition, and it is usually seen in blood vessels, usually as a result of some sort of immune response. Okay. Now here, this is fat necrosis, and this chalky white area is fat necrosis with underlying calcium deposition. That process is called saponification. Fat necrosis you see in acute pancreatitis and in lesions and in condition that has something to do with mesentery, where there is an increase in adipocytes. Now coagulative necrosis is seen in liver. Now breast necrosis in the breast happens as a result of infection. And the breast tissue does not undergo ischemia or something like that. In breast, if there is a decrease in something, either it is a result of infection, in which case it is liquefactive, or breast will undergo apoptosis. Never necrosis. Okay. Now, an example of physiological atrophy is, now I said earlier, you have your normal cell homeostasis that is maintained. Then you have some sort of stress in which the cell adapts. The cell adaptation can be of 4 types. You have hyperplasia, which is increase in number. You have hypertrophy, which is increase in size. You have metaplasia, where one type of cell is replaced by another, and you have atrophy, that is the cell is shrinking. Now, in atrophy, the most classically described physiological atrophy is uterus post delivery. Now, in delivery, what is happening in gestation, you have both uterine hyperplasia and hypertrophy. But hypertrophy means that all these muscle fibers in the uterus, they become big in size, right? So after partition has taken place, there is no use of these big uterine fibers in the body. So it will undergo atrophy. Now, senile atrophy is what happens as a consequence of age. Disuse atrophy is when an organ, or more classically when a muscle is not used, it will undergo atrophy. Now, why should it undergo atrophy? If a muscle is present in its normal state, it is taking up some nutrients and some oxygens from the body. If that muscle is not getting used, the body thinks, why should it spend that much amount of nutrients and oxygen on that cell? There is no point. So, that type of muscle undergoes atrophy. Now, it is most commonly seen either in old age, when in old age when people are bedridden or something like that, they are not using all their muscles properly, and those muscles will undergo atrophy. That is your disuse atrophy. Also, if you are bedridden for a long amount of time because of some fracture or something, there is a chance that that muscle can undergo atrophy. To prevent that, we do some sort of exercises and then we do some position changes and all that. But again, the reason why this is doing is the body is thinking there is no point in spending that much energy and that much oxygen on a muscle or an organ that is not required at present. So the body will withdraw all of that, or withdraw all that organs and oxygen and nutrients, resulting in atrophy. Now here you have another figure. This is the brain of a young adult, and this is the brain of an old person. So what you can see is you have sulci and gyri. You have wide sulci. Gyri are the projections and sulci are the crypts. Okay, so these sulci are widened and these crypts are narrowed. Okay, so that is all about atrophy. Now, unfolded protein metabolism is associated with. So here, we are going to talk about endoplasmic reticulum. So in endoplasmic reticulum, what is the function of endoplasmic reticulum? Endoplasmic reticulum is a structure like this, usually situated next to the nucleus, and it has so many ribosomes on its surface. And it is usually in close proximity with the Golgi body. So its primary function is production of proteins. Now suppose, usually what is happening is, there is a protein in the ER. Normally it is present properly and the ER will send it out. But suppose this protein is folded, that is, there is some defect in the structural integrity of the protein. What is going to happen? These structurally deficient proteins cannot be released by the endoplasmic reticulum to the cytoplasm. So it will accumulate in the endoplasmic reticulum. As a result of that, you will have accumulation of misfolded proteins in the endoplasmic reticulum, as a result of which you have something called ER stress. That is, the endoplasmic reticulum is under stress. Now, the endoplasmic reticulum will try some adaptative methods. If it is not working, it will trigger apoptosis. So essentially what is happening is, misfolded protein or unfolded protein. First, the endoplasmic reticulum will try to adapt and correct the protein structure. If it is not working, the cell will have to undergo apoptosis. This process is called ER stress response. Again, you have a figure in which you have misfolded proteins, then it will reduce protein synthesis, increase protein degradation. These are the adaptive changes that the cell will try to do. Increase number of chaperonins. Now chaperonins are proteins that help in proper alignment of this, are proteins that help in proper alignment of the misfolded proteins. Now this will have reduced load of misloaded protein. But if that is not working, there will be activation of caspases and apoptosis of the cell. Why is this important for you? It is important because there are a few conditions in which misfolded protein causes diseases. Now, most common one is cystic fibrosis, that is because of CFTR protein. Then you have your familial hypercholesterolemia because of LDL receptor. Then you have your Tay-Sachs disease because of hexosaminidase beta subunit. Then your retinitis pigmentosa as a result of rhodopsin, and your alpha-1 trypsin deficiency. Okay. Now that is all about ER stress response. We'll move on to the next question. True about metastatic calcification is. So here we are dealing with calcification. What is the meaning of the word calcification? Calcification refers to abnormal deposition of calcium in addition to something like your iron, magnesium that takes place in tissues. So calcification means essentially calcium is getting deposited in the tissue. Along with calcium, some amount of iron and manganese and some minerals are also getting deposited. It is of two major types. You have dystrophic calcification and you have metastatic calcification. Now dystrophic calcification, D for dead and dying tissue. Here the calcium level will be normal. You will have normal calcium level, but because the cell is dead and dying, there will be deposition of calcium in that kind of cells. So the cell, so in microscope, or it is going to be fine white granules that are deposited in dead and dying tissue, and when you are trying to cut such a tissue, you will have a gritty feel. Now in metastatic, as the word suggests, there is some problem in the normal calcium metabolism in the body. As a result of that, there is a increased calcium level, or rather than increase, we can say there is an imbalance in calcium level in the body, and as a result of that, calcium gets deposited. Now there are 4 conditions which cause an increase in calcium in the body. The first one is when the parathormone level increases. There is hypercalcemia. Second one is when there is increased resorption of bones. Now this increased resorption means the bone gets broken down, as a result of which you will have an increase in the calcium in blood. Then third one is some vitamin D related disorder. Then fourth one is renal failure. Now in renal failure, let me add a page. Fourth one is renal failure. In all these conditions, there is hypercalcemia, and as a result, calcium gets deposited in tissue. Now, it is usually deposited in your interstitial tissue, like your gastric mucosa, your kidney, your lung, then your arteries, and then your pulmonary vein. So again, coming to calcification. Calcification refers to the abnormal accumulation of calcium in the tissue. It is not just calcium. In addition to calcium, you will have some iron, manganese, and other minerals that are also deposited inside the cell. It is of 2 types. You will have dystrophic calcification and metastatic calcification. In dystrophic calcification, the calcium levels are going to be normal. D for dead, D for dystrophic. So in dead and dying tissue, in case of normal calcium levels, some amount of calcium is getting deposited, and it is seen as a white, fine, granular material which is gritty to touch. In metastatic calcification, the calcium level is abnormal. So as a result of that, there is deposition of calcium in interstitial tissue like your gastric mucosa, your kidney, your systemic veins, etc. And that can be because of 4 reasons. The hypercalcemia can be because of 4 reasons. First one is it can be because of increase in parathormone. It can be because of increased resorption of bone. It can be because of renal failure, and it can be because of vitamin D related causes. So as a result of all of that, calcium is getting deposited. Now, how do you see calcium in tissue? So calcium in tissue, in normal H and E, that is your hematoxylin and eosin, calcium is deposited as a basophilic, that is a purplish hue purple colored amorphous material. Now, two stains for calcium. You have your von Cosa stain and your alizarin red stain. Now, von Cosa stain will stain calcium black. In alizarin red, as the name suggests, calcium is going to be stained a reddish orange color. Now, another thing about calcification you need to learn is there is something called a Samoma body. Now, the Samoma body is essentially a condition in which calcium gets deposited inside cells, inside tissues with some neoplastic change. So, it will have a lamellar structure. That is, you will have a purple structure like this and you will have lamellations. Now, conditions in which Samoma bodies are seen are papillary carcinoma thyroid. This is very important for your exam. Samoma bodies usually seen in papillary carcinoma thyroid, then papillary renal cell carcinoma, then in your meningioma, then in prolactinoma, then in serous cystadenoma ovary. Okay. So here the question was, true about metastatic calcification is calcium level is normal. No. Occurs in dead and dying tissue. No. Occurs in damaged heart valve. All of these is dystrophic calcification. Dystrophic, D for dead, D for dying. Okay. Next question. So here you have a very beautiful picture in which there is some substances that are accumulated in the cell. That is some intracytoplasmic or some intracellular accumulation. Now imagine, if you see the first picture, you have accumulation of fat. That the word is called steatosis, and usually the fat that is deposited, it can be either triglyceride or it can be cholesterol. Now in cholesterol, it is usually deposited in conditions called xanthoma. Like you will see in some patients with hypercholesterolemia, you will see some yellowish color nodules on their skin, around their eyes and all. Those things are called xanthomas. And then you have atherosclerosis. Again, another condition in which cholesterol is deposited. Now, if triglyceride gets deposited, it is called steatosis. Now, similarly, you have many other things that are deposited. For example, if it is protein that is deposited of any sort, how will it look? In H&E, fats is going to be empty. Like fat deposition, you will see it as cytoplasmic emptiness, vacuolation. Now fat is for fat fixation, you need absolute alcohol, and it is most beautifully done in frozen section. So coming on to protein, you have protein deposition. Now proteins are deposited as eosinophilic granules. For example, you have a condition where in multiple myeloma, where you see some structures called Russell bodies. Now these Russell bodies are these pink colored bodies, that is this deposition of protein. Now similarly, in Alzheimer's disease, you have some neurofibrillary tangles that are deposited. Now you have hyaline that is deposited in some condition. Here it is called Mallory hyaline bodies, and it is seen in alcoholic liver disease. And again it is composed of intermediate fibers, that is your cytokeratin 8 and 18. Now another thing that is deposited is your glycogen. Again, glycogen in glycogen storage disorders. Here the tissue is fixed similar to fat in absolute alcohol, and the stain used is best carmine or PAS, per iodic shift. Now next question. Calcification of soft tissues without any disturbance in calcium is called dystrophic calcification. We'll take a small break right now. So welcome back. We were discussing intracellular accumulations. So we were initially talking about calcification, which is of two types, that is metastatic and dystrophic. And this question is actually dealing with calcification of soft tissues without any disturbance of calcium metabolism, and that is your dystrophic calcification. So we saw about accumulatory substances like proteins, which usually are eosinophilic in nature, and examples you have in multiple myeloma, you have your Russell's body, then in Alzheimer's you have your neurofibrillary proteins, then in hyaline bodies, for example in Mallory Denk bodies in alcoholic liver disease, this Mallory Denk body is composed of cytokeratin 8 and 18. Now glycogen. Again, glycogen is stored in tissues, and for visualizing such a glycogen, you need to fix the tissue in alcohol, absolute alcohol, like in case of fats, and the stains that are used is best carmine and PAS. Now, there is another topic that I want to deal with, a small topic, that is pigments. So, which are the pigments that are usually deposited in the body? Now, it can be both exogenous pigments and endogenous pigments. Now, exogenous pigments, a very common example is when you are tattooing something onto your body, the pigment in the ink is going to stay in your body forever. Now, where is it going to stay? It is either going to be in your dermal macrophages or even in the lymph node. So that is an example of exogenous pigment. But what is interesting about the tattoo ink is that it does not cause an inflammatory response even though it is a foreign substance. Nobody knows why the reason is. So exogenous substance, the first one you have tattoo, and that is stored in the dermal macrophages and also in the lymph nodes. And secondly, you have your coal dust. Your coal dust, it is also called anthracotic pigment. And it is black in color. Now, one doubt everybody has is, should a person be a smoker for there to be anthracotic pigmentation in the lungs? The answer is no. So anthracotic pigmentation essentially means that you have these black soot particles that are there in the body, and if you do an autopsy from my body, your body, or everyone's body, some amount of dust particles is always going to be there, because we live in a very polluted environment where these dust particles and these coal dust particles are all around us. So two types of exogenous pigments. The first one, you have tattoos, that is stored in the dermal macrophages and in the lymph nodes. And the next one, you have anthracotic pigments, that is stored in the lungs. And there is a condition called coal workers pneumoconiosis, where these pigmentation is these black soot particles are very prominent. So we'll move on to endogenous pigments. So endogenous pigments, the word means that it is something that is produced by the body and it is present in the body. So the most common example is your lipofuscin. Now, few points about lipofuscin. The first thing is, it is called a wear and tear particle. Then it is seen in something called brown atrophy. You see the word brown atrophy, and whatever is the pigment that is involved, the answer is always lipofuscin. Please remember. Now, it is present, as I said earlier, it is a wear and tear pigment. So, it is present in all aging tissues. And also it may be seen in cancer cachexia, and something like that. So in your normal HND stain, it is going to be some yellowish brown substance. We have a picture that is coming later. So yellowish brown substance usually seen in all aging tissue. Even in the heart muscles you can see lipofuscin, so that is lipofuscin. Second one, you don't need a separate stain for lipofuscin. In normal HND it stains some golden brown color. So the next one is melanin. Melanin is another pigment, black color pigment that is present in the body. And the stain that is used for melanin is called Mason-Fontana. Now, whenever you hear the word Mason-Fontana, there is one thing you have to remember. You have true, as some books actually say it is pronounced Mason, not Mason. I don't know. What I know is Mason-Fontana. So, Mason-Fontana is the stain used for melanin, while Mason trichome is the stain used for collagen. That is very important. Please don't confuse the two. If it is Mason-Fontana, it is melanin. If it is Mason trichome, it is collagen. Now coming on to the other pigments. Now melanin is seen in another condition which is called ochronosis. That is as a result of your alkaptonuria. Then moving on to the third pigment, again it is hemosiderin. Now hemosiderin is essentially iron in another form. So in your normal HND, it is going to be stained yellow to brown. Now because hemosiderin contains iron, the stain for iron is always pearl stain or Prussian blue stain. Okay, that's all about endogenous pigments. So pigments, you have two types, exogenous and endogenous. Exogenous, you have your tattooing pigment that is present in your dermal macrophages and in your lymph nodes. Then you have your anthracotic cold pigment that is present in the lungs. Coming to endogenous pigments, the first one is lipofuscin, which is the wear and tear pigment, and it gives a brownish, yellowish brown color in normal HND. It is seen in brown atrophy and also in aging tissues. Now, melanin is another pigment which is black in color. The stain for melanin is Mason-Fontana. Remember, Mason trichome is for collagen and Mason-Fontana is for melanin. The other pigment that you usually see is hemosiderin, which is essentially iron, and it looks somewhat yellowish in HND smear, and the main stain that is used for iron is your pearl stain or your Prussian blue. Okay, now moving on to the next question. Transformation of one epithelium to other epithelium is known as metaplasia. So here, I said earlier, you have cell, some injury or some stimuli, you have adaptation, and adaptation, you have several types of adaptation. You have hyperplasia, hypertrophy, then you have atrophy, and then you have metaplasia. So metaplasia is the replacement of one type of cell by some other type. Now, the most commonly given example of metaplasia is your Barrett's esophagus. Now, what are the other words that are used here? Now, before we go on to that, there are a few words which you need to know. First one is hamartoma. So, what is a hamartoma? So, hamartoma is a collection of cells disorganized cells in normal site. That is collection of disorganized cell in normal site. The cells are not metaplastic or dysplastic by any means. They are normal cells, but they are not properly arranged and they are usually seen in their normal site. Now, choriostoma or heterotropic tissue is one in which you have normal cells in abnormal location. That is choriostoma or heterotropic tissue. Now, what is dysplasia? Now, dysplasia means there is some abnormal change that is taking place in the cell. As a result of dysplasia or as a marker of dysplasia, what usually happens is you have the cell. When do you call the cell dysplastic? When there is an increase in NC ratio, that is the nucleocytoplasmic ratio. When there is a there is a pleomorphism, that is change in size between cells. Then change in shape, all of those cells is called dysplasia. Now, other word I want you to know is differentiation. So, what is differentiation? Differentiation is you have your human embryo. Embryo has so many stem cells. Now, stem cells, the word stem cell means that it can become any cell. That is, it can form a mesodermal cell, it can form an ectodermal cell, it can form an endodermal cell. That is, it can be a neuron, it can be a muscle cell, it can be an endothelial cell, it can be a neutrophil. But once the stem cell goes into a progenitor mode, you have, for example, we'll take the example of RBCs. So you have stem cells. Now stem cells is going to form something called common RBC precursor cells or progenitor cells. Now once this cell becomes progenitor cell, it cannot go back to its stem cell position. These cells can differentiate. They can differentiate into RBC alone. I hope that is clear. That is the difference between stem cells and progenitor cells. So, again you have this RBC that is forming blood cells through a very big process. Similarly, suppose you have some endothelial cell. Endothelial cells are those cells that are lining your blood vessel, right? So what is happening? Suppose you have a cell that is giving rise to an endodermal progenitor cell and it is giving rise to an endothelial cell. Now how much does this endothelial cell, that is this endodermal progenitor cell giving rise to endothelial cell, how much does it look like a normal endothelial cell? That is how much that does the tissue that is formed look like the original tissue that it is supposed to form. How much does the tissue that is formed is similar in size, shape and function to the tissue it is supposed to form. That process is called differentiation. So if you have a muscle cell, the muscle cell progenitor that is giving rise to a muscle cell, how much does this muscle cell appear like a normal muscle cell? That is called differentiation. And loss of differentiation is called anaplasia. I hope that's clear. So you have, coming on to the question, you have transformation of one epithelium to another epithelium, and that is called metaplasia. We have seen what is dysplasia, hyperplasia, neoplasia. Neoplasia is essentially malignancy. Okay. Let's move on to the next question. Oncocytes are modified forms of which of the following? The answer is mitochondria. So, oncocyte. What does the word oncocyte mean? I will explain that in terms of thyroid. In thyroid, this is the structure of thyroid. You have follicles that are colloid filled, that are lined by follicular cells. You have follicles that are lined by follicular cells. Suppose a follicular cell, this is how it looks like. When you see it in microscopy, you see some sort of small cell with a very prominent purple nuclei, basophilic nuclei, and little to moderate amount of eosinophilic cytoplasm. Now suppose this eosinophilic cytoplasm increases. The amount of this eosinophilic cytoplasm increases, and this pink colored eosinophilic cytoplasm acquires somewhat of a bluish hue. Now, when I say pink and bluish, you just have to understand that this pink colored thing, it is gaining some sort of granularity. So now you have a cell with a bigger nucleus and some sort of granularity and an increase in cytoplasm. Those kind of cells are called oncocytes. And it is usually the mitochondria that are increased in oncocytes. Now, let's move on to the next question. Fixative used in histopathology is 10% buffered formalin. So, you have fixative. So, what do you mean by fixative? Fixative, what is fixation? Fixation is essentially you are arresting the cell in its form. That is what is meant by fixation. You are fixing the cell in that form. Suppose you are doing a thyroidectomy. You have taken the thyroid out from the body and you need to know what is the pathology in that thyroid. Now, once the organ is coming out of the body, it is going to undergo some changes because the normal nutrient supply, the oxygen that is keeping this thyroid alive is no longer there. But we need that cell like that because it is only then that we can understand what is the pathological basis or what are the changes in the pathology of the normal thyroid that is giving rise to a condition, whatever it may be. So essentially, you need to make sure that the thyroid is preserved in a beautiful condition so that once we section the specimen, once we get slides from the specimen, it is properly maintained. Now that process in which you are properly maintaining the cellular integrity and the cellular conditions in an optimal way is called fixation. And the substance you use for fixation is called a fixative. Now, what you need to know is histological fixative. In histology or histopathology, the most common fixative that we used is 10% neutral buffered formalin. For electron microscopy, the common fixative used is glutaraldehyde. Now, for cytology slide, you can use many kind of fixatives. You can use carbovax, many kinds of fixatives are there. The most commonly used ones are carbovax and isopropyl alcohol. And then you have something called your peripheral smear. I hope all of you know what a peripheral smear is. Essentially, you take a drop of blood, then you smear it on a glass slide. That is a peripheral smear. And it will give a good look at how the hematological system is working in your body. So what do you think is the fixative used in peripheral smear? Peripheral smear does not use a separate fixative because the fixative is inbuilt in that stain, inbuilt in the stain. So you don't need any fixative because the fixative is inbuilt in the stain. I hope that is clear. So, most common use of histochemical fixatives, you have formaldehyde, glutaraldehyde. Glutaraldehyde is used for electron microscopy. You have vapor fixatives. Then all of these are other fixatives. Cytological fixative, you have glacial acetic acid, alcohol, formal saline, all of these. The most commonly used one is either carbovax or isopropyl alcohol. Now, you have, as I said earlier, let us take the condition. You have a thyroid tissue that you have a thyroid that is taken from the body. So what do you do with this thyroid? I said earlier, the first step is fixation. And second step is, finally, you need histochemical slides. That is, you need to take a bit from that thyroid, a bit from the abnormal part of thyroid, and you need to study it under microscope to understand what is the change that is taken place. So here you have an organ that is taken from the body, and finally you need a slide. And in that slide you will have a bit of tissue. So what are the processes that is taking place? Just in short, I will tell you. One, you do fixation. Then you do something called grossing. So essentially in grossing, what is happening is you are taking that thyroid, which is a very big specimen, and you're thinly making slices out of that thyroid. Why should you do this? This is to make sure that the fixative is able to penetrate the whole of tissue. Just imagine you take a ball, you take a ball, a sponge ball, you put it in water. Once you put it in water without cutting, or you put it in water with cutting, naturally if you are putting it with cutting, the rate of penetration will be more. The rate of penetration of water into the sponge will be more. Similarly, in the case of tissues, you are grossing it first, you are making thin slices so that penetration of fixative can take place properly, and after that what you are doing is you are taking a small bit from that slide, and then this thyroid, you are taking a small section from that thyroid, and the small section what you are doing is you are embedding it in paraffin wax to preserve the architecture, and then you are taking a thin slice of that tissue, and then you are staining that tissue, and then you are analyzing that tissue. This is what is happening in histopathology. You take a body, you take a specimen, whether it be your uterus, your thyroid, even your small biopsy, your true cut biopsy, whatever it may be. First, you have to make sure it is ideally fixed. For fixing, you do something called grossing, in which you are making sure that the fixative is able to completely penetrate the tissue. And after that, you do sectioning, and from that sectioning, sectioning means you are making small sections from that big thyroid. So that big thyroid, from the small section I mean ideally it is only 1 into 1 centimeter. You are taking that small bit of tissue, you are embedding it in something, usually paraffin wax or something, and you are taking a slice out of that tissue and you are analyzing it under microscope. That is what is happening in pathology. This is just for your knowledge. So the topic here was fixatives. So all of these things, fixatives, have special uses. In histology, the most common fixative is your formaldehyde. In cytology, you can use your carbovax, then you can use isopropyl alcohol, you can use many kinds of fixatives. And in your hematology, in routine peripheral smear, you are not using a fixative because the stain itself has an action of fixation. Okay. Now coming on to the next question. Which of the following is a paroxysmal free radical scavenger? Okay, so here we are dealing with another topic. We are dealing with free radical injury. Now, what is a free radical? A free radical is any compound or any molecule that has a single free electron in its outer orbit. Okay, so free radical, you have a single free unpaired electron in its outer radical. If there is an increase in this free electrons inside a cell, it causes something called oxidative stress to the cell. Now, as a result of this oxidative stress, the cell will try to scavenge or use up the free radicals, and if it is not able to use up the free radicals, it will undergo cell injury and ultimately cell death. Now, how does the body remove or scavenge these free radicals? Now, for free radical removal, first point is with the use of antioxidants like your vitamin E, your vitamin A, your glutathione, then your ascorbic acid, that is vitamin C. All of these are free radical scavengers, and they take up the extra free radicals. If you are interested in skincare of any type, you will see that most common marketing ploy that people use is it is rich in antioxidants, and hence it will take up these reactive oxygen species that are there in the body, that is free radicals. And example for that is you have your vitamin A, you have your vitamin E, glutathione, and vitamin C. Now, second is with the help of ion compounds, minerals like your iron, your copper, your magnesium. All of this can also take up electron. Then third is through some enzymes. Here you have some 3 main enzymes. The first one, you have catalase, that is present in your peroxisome usually. Then you have your superoxide dismutase. which is of two types. That is, you have your copper zinc superoxide dismutase that is in your cytoplasm, and you have your manganese superoxide dismutase that is in your mitochondria. M for manganese, M for mitochondria. Then you have another thing called peroxidase. These are the normal, most common three enzymes that deal with peroxisomal free radical injury. So, in this question, which of the following is a peroxisomal free radical scavenger? All are the answers. I mean, all three are answers. They are all present everywhere. So here you have a mitochondria. As you can see, there is radiation, toxins, reperfusion that is causing the production of ROS. Now, some sort of ROS can be scavenged by the body. If the scavenging is not taking place properly, you have three effects. The first one is you will have something called lipid peroxidation. That is, you have the plasma membrane that is composed of phospholipids. It gets broken down and that causes your membrane damage. That is your first point. Second point is, because of these free radicals, you have some protein degradation that is taking place. That is, it can be protein breaking, misfolding, etc. and that can cause cell injury. And the third way is through DNA damage. That is, free radicals can cause mutations. That is all about free radicals. Now here you have a table in which the most common free radicals. Now this I will tell you, please go through this table very carefully, because your question is going to be which of the following is a free radical, and you will have something like OH minus, NO minus, NO dot, NOO dot, things like that. So you need to know what are the proper methods of depicting these free radicals as well. So the first one is O2 dot minus, then you have H2O2, then you have ONOO dot. That dot represents the extra electron. And here, as you can see, it can have damaging effects on lipids, proteins and DNAs. Now coming to the next question, this is a very important question. Stain used for melanin is Mason-Fontana stain. So staining, as I said earlier, you have your tissue, whatever tissue it is that is taken from the body. You fix the tissue undergoes some sort of processing, and after that it gets embedded, and after embedding it gets stained and then analyzed. For different materials inside the cell, different stains are required. So please write this with me. First one, amyloid. So amyloid is a protein that is present, and a very commonly asked question. It is stained by Congo red and under polarized light. That word is very important. This amyloid, when you are staining it with Congo red, if you are looking under normal microscopy, the only thing you are going to see is this pink eosinophilic material. Once you look at it under polarized light, you have something called apple green birefringence. Okay, amyloid. Other stains for amyloid include your theophlavin S. That's all. Now carbohydrates. Your most common ones are Alcian blue, then your PAS, that is periodic acid shift for carbohydrates. Then your triglycerides and lipids, you have oil red O and Sudan black. Then for your reticulin fibers, you have reticulin stain. Then I hope you remember, as I said earlier, for your melanin, you have what is called a Mason-Fontana stain. Then for your collagen, you have Mason trichome stain. Then you have for iron, you have your pearls stain. Then for calcium, you have your von Cosa stain and your alizarin red stain. Here, as you can see, you have amyloid, congored. Then for mucin, especially acidic mucin, you have your Alcian blue. Here, if you can look properly, you have here the more important ones, because you have so many stains for so many things. Another thing, for hematological specimens, that is for your peripheral smear, you use something called a Romanowski stain. That is, you use either your Leishman or your Giemsa. Now this Romanowski stain, what you mean by Romanowski stain is that the stain will have an acidic component and a basic component, and so the basic part of that will basic part of the dye will bind to the acidic part of the cell, and the acidic part of the dye will bind to the basic part of the cell. You don't have to worry about all of that. Hematological stain most commonly used is your Romanowski stain, that is your Leishman and your Giemsa. So that is all about stains. Before we move to the next topic, we'll have a 1 hour lunch break. So we'll be back after 1 hour. So hi everyone, welcome back. Hope you all had a good lunch. Sorry for the delay. We had a small technical issue. So characteristic of acute inflammation is. Now we are moving on to the next topic, that is inflammation. Now what is inflammation? Inflammation is a process by which the cells it is a process by which the cells respond to whatever stimuli that is there and how the cell tries to overcome that stimuli. Now, in inflammation, you have 5 R's. That is, you have recognition, then you have recruitment, then you have removal, then you have regulation, and then you have repair. Again, here, as it is beautifully shown in this picture, you have, first, we need to recognize that there is something wrong in the body. After it is recognized, so here as you can see, you have a microbe and you have some antigen presenting cells, which may be the dendritic cell, the mast cell, whatever it may be. It is recognizing the fact that there is something wrong or something noxious that has come into the body milieu. Then after that, the second step is recruitment. Now recruitment is, there is recruitment of other cells, like you have your neutrophils, you have your monocytes, your macrophages. Now how is this recruitment done? This recruitment is done via some mediators. Now once the cell has once the body has recruited all the cells that are necessary for inflammation, the third point is removal. Now removal, as the word suggests, as you can see here, the whatever the thing may be, maybe it is a microbe, maybe it is a toxin. The body has done some mechanism to overcome that noxious stimuli and to remove that noxious stimuli. That is removal. Now fourth thing is, you need regulation. Once that noxious stimuli has moved on, the cells have to be brought back to its normal, that is to its normal state. That now, because of that noxious stimuli, all the cells have been activated. So you have activated neutrophils, macrophages, monocytes and all those cells. Now these have to be brought back to the original position. That is called regulation. And after that, the last step is repair. Now repair, what do you mean? In repair, once we understand that there are some things that can be replaced and some things that cannot be replaced, and what cannot be replaced has to be repaired. That can occur by primary intention or secondary intention. We'll be dealing with that later. So in short, in inflammation, you have recognition, recruitment, removal, regulation and repair. Now here is another table that gives the difference between acute and chronic inflammation. Acute inflammation, as the name suggests, you have fast response. In chronic, it is days, that is slow when compared to fast. Then cellular infiltrate is mainly neutrophils in case of acute inflammation and monocytes and macrophages in case of chronic inflammation. In tissue injury, in acute inflammation, it is usually self-limited, while in chronic, it may be severe and progressive. Now, local signs are present in case of acute inflammation and absent in case of chronic inflammation. So here, as you can see, what was our question? Our question was, characteristic of acute inflammation is. So, as I said earlier, once the body recognizes that there is some noxious stimuli, some adverse event that has adverse event or some adverse factor that is there in the body has to recruit cells into that position. For cells to be recruited into that position, what do we need? You have your cells Suppose this is a microbe and here you have your blood vessels. Now you need to recruit all these monocytes and macrophages and everything. Where are those kind of cells present? They are present in the blood vessel, right? So from the blood vessel, these cells have to migrate from the blood vessel or wherever they are formed. From there it has to come to the site of injury. Now for that, in usual parlance, what you see is you have endothelial cells that are covering the blood vessel. Now there has to be an increased permeability, that is these endothelial cells has to retract so that the blood cells, that is our leukocyte, monocyte, macrophages, everything, they have to come from the blood vessels where they are present into the site of injury, that is to the tissue. So the first step is always an increase in vascular permeability. So that is the answer for this question. Now we'll see the rest of acute inflammation that is taking place. So here, as you can see, you have the normal what is happening normally in the body. You have the normal tissue, you have the endothelium, and you have the blood. Now, this endothelial cells, they have to retract. What does the word retraction mean? So, if this is the cell, suppose the cell is this big, these cells have to retract, that is they have to make space for the blood vessel, for the blood cell, that is the leukocyte, to come from the blood into the tissue. So here, as you can see, these cells, they are retracting. As a result of this retraction, all your blood and your plasma proteins, and these small things are your plasma proteins, then this is your neutrophils, so that all of that things can come from the blood vessel into the site of injury. Now, suppose there is an endothelial injury that is severe or of long-standing duration, then what is going to happen? Then these cells, they will contract. Now, retraction is a small transient process. What does the word transient mean? That is, it lasts for a second, for few seconds, for few moments only. That is transient. But once there has been endothelial injury, because of whatever the cause may be, maybe it is because of infection, maybe it is because of burns, because of toxin, the cells contract. That is, they permanently change into another position. So they contract. So that is caused by burns, microbial toxin, and here it is long lived, that is it may take place, that is it may last hours to days. Okay, so here I hope that much is clear. So next question, that is what is asked in the next question. In acute inflammation due to contraction of endothelial cytoskeleton, which of the following results? You have an early transient increase in the permeability. Okay, so that is there. So here you can see early transient increase is because of endothelial cell contraction, and you have a delayed transient increase, that is because of endothelial injury, and delayed permanent increase, that is because of damage. I hope that much is clear. Now we'll move on to the next question. So now we have seen, now we are going on to acute inflammation, the topic proper. So now where have we reached? You have tissue with injury, and you have a blood vessel which has increased permeability, and because of which the neutrophils need to come out. So inside the blood vessel, all these neutrophils and the lymphocytes and the blood vessels, they occupy the central portion, and blood flows in the periphery. Because these are proteins, they occupy the central portion. So from the central portion, the leukocytes, that is the neutrophils, we'll call them leukocytes because it may be cells of any types. It may be a neutrophil, it may be a lymphocyte, it may be a macrophage, it may be a monocyte. Whatever the cell may be, the cells are present in the center of the blood vessel. These have to move on to the periphery. That process is called margination. Then secondly, once they have come to the margin of the blood vessel, what do they have to do? They have to reach and they have to adhere to the endothelium so that they are able to permeate, so that they are able to migrate through the endothelium into the tissue. So the second step, you have rolling. Then the third step, you have adhesion. Then the fourth step, you have something called transmigration or diapedesis. So in this picture, it has been beautifully given. So here you have, I hope you can see, you have endothelium. So here first all these leukocytes that are in the center of the blood vessel, these are rolling, and then they have to adhere to the endothelial lining, and then after that, through this break in that endothelial lining, these have to transmigrate. This process is called your transmigration. Okay, so here you have some factors that help. You have some factors that help in identifying and activating these neutrophils. So for that, the first step for that activation, you have proteins called selectins. You have three types of selectins. You have P-selectin, E-selectin and L-selectin. Now P-selectin is present on the endothelium, and P-selectin is present on the endothelium, E-selectin is also present on the endothelium, and your L-selectin is present on the leukocyte. So for that activation, your selectins have a role. Now the second thing that has to happen is adhesion. See, you can see here, for this adhesion, you have some proteins called integrins. For example, you have some proteins for VCAM, ICAM and all. All of these help in adhesion. Now the third thing is you need transmigration. For transmigration to occur, you have a protein called PCAM, also called CD31. I hope that is clear. Now this is what is happening in acute inflammation. So moving on to the next question, function common to neutrophils, monocytes and macrophages is phagocytosis. So earlier when we learned that in acute inflammation, neutrophils are the primary cells that are linked. Then in chronic inflammation, you can have monocytes, lymphocytes and macrophages. So here you have a table which gives the difference between neutrophils and macrophages. Now neutrophils, both neutrophils and macrophages originate in the bone marrow. Here when you can see, you have a common white blood cell precursor. They form two types of cells, that is granulocytes and agranulocytes. Now your granulocytes include your neutrophils, eosinophils, basophils. Agranulocytes include your monocyte, your macrophage, then your lymphocyte and all. So neutrophils, the lifespan is 1 to 2 days, while macrophages can live for years or can live for days and even years. Response to activating stimuli, short-lived. Neutrophils are short-lived response, while macrophages have a prolonged response. Now, what is important is there is secretion of lysosomal enzymes from neutrophils. As I said earlier, these neutrophils, they are granulocytes. That means they have some granules within them, and all these granules contain lysosomal enzyme that result in breakdown of the whatever the tissue that is that the neutrophil is trying to break down. Okay. So here you have a picture. Here you have, I hope you can see here, if you look closely at this picture, you can see cells with multi-lobed nuclei. These are your neutrophils. On the other hand, here you can see cells with a single nuclei. That is your monocyte. I hope that's clear. Again, here as you can see, activity, the first thing we said, what has to take place? Increased in vascular permeability has to happen. That is, this increase in vascular permeability can cause edema. What is edema? Edema is collection of tissue, collection of fluid inside the tissue. Now, neutrophils, you have the primary response, and you have other cells, monocytes and macrophages, which are concerned with the secondary response. Okay. Now we'll move on to the next question. Lewis triple response is caused due to, the answer is histamine. So here, this triple response, what does that mean? So suppose you take a sharp object, not a very sharp object, you don't want to cause injury, you take some somewhat sharp blunt object and you suppose you draw a line in your skin. The first thing that is going to happen is there is going to be a red line. That red line is because of capillary dilatation. That is, once you have done some sort of injury, the first response, that is the first response is a red color, and that is because capillary dilatation occurs, so RBCs come there. Now after some time you can see there is something called a wheal. So here what happens is, now because all these red blood dilatation of capillaries occurs, red blood cells come out. What is left is the fluid. What is left is the fluid. That fluid is what is exudates from the blood vessel. So the second thing is you have something called a wheal. Third thing is you have a flare. Now here, because this is localized, we learned earlier, once the first step is recognition. As a part of recognition, it has an increase, there is an increase in vascular permeability that is seen by that red spot. And then because the red blood cells have escaped, you have something that is the exudates that are left. That causes the wheal response, that is the white color. Then now that the body has recognized there is something going on in that place, what is going to happen? Recruitment is going to happen. So everything has to come to that phase, to that location. For that, something called a flare response is there. That is, surrounding becomes red, because from surroundings you have all these increased blood flow and these capillaries. So that is about Lewis triple response. Now we are moving on to a very important part of your topic of acute inflammation, that is your complement system. Now I am sure it has been dealt with very well in micro, but I'll tell you something about cytokines. So what are cytokines? Cytokines are essentially, what are they? They are chemical mediators. So these constitute about 20 proteins named from C1 to C9. So here you have three pathways. So first one, you have what is called the classic pathway, then you have what is called an alternate pathway, then you have what is called a mannose binding pathway. So in very short, I will tell you what is happening. So you have the first thing, that is you have something called a C1QR2S2. That is just a cytokine complex. That is all you need to know. Now the cytokine complex, when it binds to some receptor, it is going to get activated. Now how is it getting activated? You have C4 cytokine that gives out C4A. You have C2 cytokine which gives out C2B. Now here, as you can see, what is left? You have C4A is given out, so C4B is left. C2B is given out, so C2A is left. Now this is what is called a C3 convertase. Okay, and this C3 convertase, that is where this all these pathways meet. So after that, what is going to happen? Now we are talking about the classic pathway. So here C3 convertase means it has to convert C3 only. So you have C3, you have C3A that is given out. So what is left here? You have C4B, C2A, C3B. This is called a C5 convertase. So what does a C5 convertase do? It has to break down C5. So you have C5 here that is broken down and C5A is left off. So what is left? You have C5B left. Now this C5B, it will go and bind to other cytokines such as your C6, your C7, C8 and C9. And these together will form what is known as MAC, that is membrane attack complex. Now this structure will form some pores in the membrane of the antigen and causes autolysis of that antigen. Okay, so this is the first pathway, that is, I'll take another page here. This membrane attack, this causes pore formation in antigen. Now this is the classical pathway. Now in alternate pathway, what is happening? Here, as you can see, in alternate pathway, this C1, C4 and C2 are not taking place. Directly, the pathogen is activating your C3 convertase. That is your alternate pathway. And in lectin binding, that is in mannose lectin binding pathway, your C1 is not taking place. C1 is not getting involved. So you have C4 and C2 that is going directly onto C3 convertase. So here, some questions that you have to know of is C3 convertase, that is the common point of all these pathways. So complement system, you have three pathways. You have the classic pathway, then you have the alternate pathway, then you have the mannose binding pathway. So in classic pathway, what is happening? You have a molecule called C1Q. Now C1, C4 and C2. The details you don't need to know. C1, C4 and C2. They are together forming something called a C3 convertase. Now the C3 convertase is forming a C5 convertase and it is also forming a membrane attack complex. So this thing you have to notice, in alternate pathway, the C1, C4 and C2, C1, C4 and C2 do not enter. In Mannose pathway, C1 does not enter. Now, one of the most important questions is, there is an enzyme called C1 esterase, and deficiency of that C1 esterase causes what is called hereditary angioneurotic edema. Then another few things that you need to know is, C3 deficiency causes an increase in pyogenic infections, and your C5 to C9 deficiency causes Neisseria infection. And there is another protein, that is CAP, CD59 associated protein. Deficiency causes paroxysmal nocturnal hemoglobinuria. Okay, so that is what you need to study about complement pathway. Again, another thing, this complement pathway can form something called MAC, that is your membrane attack complex, and that will cause cell lysis. Then all these things that are left, that is you have your C3A, you have your C5A and you have your C4A, all of these, that is your C3, C4, C3A, C4A and C5A, these all act as anaphylotoxins. Okay. Now, so the first question was, both antibody dependent and independent complement pathway converge on which complement? The answer is C3. Everything converges on C3. The next question is, cytokines include interleukins, are produced only in sepsis, or polypeptides have highly specific action? The answer is, it includes interleukins. So I will give you a short list of things that have been especially mentioned in Robbins, because to learn about cytokines you'll have to learn about a lot. There are many things you need to learn, but what is important is what I am writing down now. So you have first function, that is vasodilation. Now, vasodilation is mediated by histamine and also by your prostaglandins. We'll write PG for short. So your second one, you have increased vascular permeability. That is mediated by your histamine, then your serotonin. Then earlier, when we were talking about the complement pathway, we were talking about something called C3A and C5A. Those also increase vascular permeability. Along with that, you have your leukotriene, C4, D4, E4. Now, next function is chemotaxis. Now, chemotaxis, what is chemotaxis? Chemotaxis is a process by which, a microbe or a cell is attracting leukocytes towards that cell. That is, essentially, it is attracting other cells. So that process is called chemotaxis, and that along with leukocyte activation mediated by, you have TNF, which is your tumor necrosis factor. Then you have your interleukin 1. Then you have, again, you have your C3A and C5A. Then you have your leukotriene B4. One thing to remember here is your leukotriene C4, D4, E4. These always result in increased vascular permeability, whereas leukotriene B4 is concerned with chemotaxis. Now, fourth one I have to tell you is about, your fever causing interleukins. Main one is your interleukin 1. Along with that, you have your tumor necrosis factor and some prostaglandins. Now, fifth one is pain. Mediating pain, you have your prostaglandins, then you have something called bradykinins, and then you have your substance P. So again, vasodilation, you have histamine and prostaglandin. Then increased vascular permeability, you have your leukotriene C4, D4, E4. Along with that, you have histamine, you have serotonin. Then chemotaxis, you have tumor necrosis factor. Along with that, you have leukotriene B4. Then fever inducing, three things. You have your TNF, that is your tumor necrosis factor. Along with that, you have your interleukin 1 and your prostaglandin. Now, pain is mediated by your prostaglandin, your bradykinin and your substance P. Okay. So, moving on to the next question. Histamine causes. The answer is vasodilation. Again, just go through that table. The important ones I've already highlighted. In inflammatory process, prostaglandin E1 and E2 causes. Again, the answer is vasodilatation. In this figure, that alone is given. Vasodilatation, you have prostaglandins PGI2, and along with that you have PGE1, PGE2 and PGD2. Okay. In acute inflammation, a few more factors that are involved. In acute inflammation, you have your TNF, tumor necrosis factor. Then you have your interleukin 1, that is fever causing. If you remember from earlier, this interleukin 1, that is this fever causing interleukin 1, is also involved in your pyroptosis. Okay, so then you have interleukin 6, that is involved in your systemic effects of acute inflammation, and then you have interleukin 17, which is involved in recruitment of neutrophils, monocytes and macrophages. Now these are the interleukins that are concerned with acute inflammation. When it comes to chronic inflammation, you have some other things, that is you have interleukin 12, then you have interferon gamma, and you also have interleukin 17, which leads to macrophage activation. So, we'll move on to the next question. Which of the following factors is morphogenic as well as mitogenic? So, what does the word mitogenic mean? Mitogenic means ability to cause formation of cells. That is mitogenic. Morphogenic means ability to cause differentiation. The answer is your fibroblast growth factor. Okay, now before we move on to the next question, you have something that you've studied in biochemistry. You have something called your arachidonic acid, that is by the action of two enzymes, by cyclooxygenase, you have something called prostaglandins that are made. When lipooxygenase acts on arachidonic acid, you have leukotrienes. So this is a very big thing that is happening. For that, what you need to know is, from cyclooxygenase you have prostaglandins, and prostaglandins PGI2. PGI2 is involved in vasodilatation and it inhibits platelet aggregation. Now you have another factor in that cyclooxygenase pathway, which is called thromboxane A2. Now how you need to remember thromboxane A2 is that it is the direct opposite of PGI2. That is, here you have vasoconstriction and you have induction of platelet aggregation. Then further down the line, you have something called your PGD2 and your PGE2. Now both of that causes vasodilatation as well as increase in your vascular permeability. So I hope that is clear. Now coming on to the leukotriene pathway, here what you need to know is you have leukotriene C4, D4, E4 that is involved with increased vascular permeability and bronchospasm. And you have your leukotriene A4 and B4 that decreases neutrophil adhesion. Okay, so in short, arachidonic acid, you have two types of enzymes that are acting. You have the cyclooxygenase pathway and the lipoxygenase pathway. In cyclooxygenase pathway, there are several steps. In that, four molecules that you need to know of. First one is PGI2 that causes vasodilatation and inhibits platelet activation. The next factor, that is thromboxane A2, is the direct opposite of PGI2. So it is going to cause vasoconstriction and it is going to promote platelet aggregation. Now coming on, you have the other two factors, that is PGD2 and E2. Now both of that will cause vasodilatation and increase in vascular permeability. That is in short what you need to know about the cyclooxygenase pathway. Moving on to the next pathway, that is the lipoxygenase pathway. You have leukotriene C4, D4, E4, which is concerned with bronchospasm and increased vascular permeability. The next thing you have there is your leukotriene A4 and B4 that is concerned with inhibition of neutrophilic adhesion. That's all. Now, systemic inflammatory response syndrome, FORSIS. So this you've already studied in your surgery classes. In short, systemic inflammatory response syndrome, two of the following. So, temperature, either it is more than 38.3 degrees Celsius or it is less than 36 degrees Celsius. Heart rate more than 90. Then your respiratory rate more than 20. Then your WBC count, either it is less than 4,000 or it is more than 12,000. Now, in addition to this, some books say that blood glucose level more than 7.7 gram per deciliter in a patient who is not diabetic, that is also included in SIRS. So essentially here it can be leukopenia, it can be leukocytosis, it can be fever, but it cannot be hypoglycemia. So that is the answer. Now, negative acute phase reactant. So this is an extremely important table. Please go through this table the day before your exam. So what are acute phase reactants? So as I said earlier, in case of an infectious stimuli, there is some sort of inflammation that is taking place inside the body. As a result of that, there are some laboratory values that will give us a clue of some serious systemic inflammatory response. It can either be and these kind of molecules that give us a laboratory indication of severe inflammatory response that is taking place inside the human body, those substances are called your acute phase reactants. It can be of two types. You can either have positive acute phase reactants or you can have negative acute phase reactants. As the name indicates, what is happening in positive acute phase reactants, if the value of that reactant is more, you can understand that there is a severe inflammatory response that is taking place. Similarly, in negative acute phase reactants, the fall, that is the lowering of that value, is also significant because that signifies there is an acute inflammatory reaction that is taking place. So you have positive acute phase reactants and negative acute phase reactants. I hope that difference is clear. So positive acute phase reactants, the examples are your transferrin, the examples are your C-reactive protein, then your ESR, then your ferritin, that in addition to whatever is given here. In acute active phase reactant, the most common indication is albumin, then transferrin, transthyretin and retinol binding protein. So we'll move on to the next question. Granuloma is a pathological feature of all except. Okay. Now, until now, we've been dealing with acute inflammation. Now we'll move on to what is called chronic inflammation. Okay. Now, in chronic inflammation, what is taking place? Acute inflammation, as we said earlier, there is increased vascular permeability. As a result of that increased vascular permeability, the leukocytes are marginating, rolling, adhesion, then transmigration, and it is coming and then it is killing the microbe and whatever. Here, what is happening is, first point, there is an infiltrate composed of mononuclear cells. So, which are the mononuclear cells? You have monocyte, lymphocyte, and even macrophage can also be called a mononuclear cell. The second thing is, as the name suggests, it is chronic inflammation. So, some sort of tissue destruction is going to happen. So, the second point is there is some sort of tissue destruction that is happening. The third point is, now once there is some destruction that has taken place, do you think the body is going to sit simply and allow that destruction to take place? No, it cannot do that. So the body is trying to compensate for that destruction in one way or the other. So you have an attempt at repair. So that is, it can be by two methods. It can either be by angiogenesis, that is, it can be by increased number of blood vessels, wherein you have some increased nutrients and oxygen and everything that is coming there to that place which has undergone some sort of damage. The second thing is fibrosis. So in fibrosis, essentially what is happening is the body has already decided that there is some damage that has occurred. Now the body wants to, it's like suppose there is a crack in the wall. The first thing you do is you try to put some cement on that crack to cover up that crack. Similarly, the body is trying to put some tissue into that damaged area to cover up that damage. That process is called fibrosis, essentially speaking. So, now chronic inflammation, the most common pattern of chronic inflammation is your granulomatous inflammation. Now, what do you mean by a granuloma? Granuloma is essentially a collection of activated macrophages and T lymphocytes. along with or without necrosis. Now it can be of two types. You can have something called a foreign body granuloma, or you can have an immune granuloma. Now, foreign body granuloma, the most important or the most common thing is, suppose a patient has undergone some sort of surgery and some sort of injury, so not surgery, injury, and then you are suturing that wound, whatever wound that is there. Now, if some suture material is left there inside the body body is going to form some foreign body giant cells near that inflammation, and that whole thing is called a foreign body granuloma. On the other hand, in immune granuloma, it is Th1 mediated. Th1 means T helper cell subset cells type 1, and you have something called your interferon gamma. Okay. So here, what is the question? Granuloma is a pathological feature of all except the answer is microscopic polyangiitis. And here, you have a beautiful table which says examples of systemic vasculitis with granulomas and without granulomas. You hear the word granuloma, what is the most common example that comes into your mind? It is of course TB. So if we are talking some examples of granulomatous inflammation, first thing is always going to be TB, and it is what is called a caseating granuloma. Caseation we studied earlier. Caseation is the formation of cheese-like material. So in TB you have a caseating granuloma. Then in sarcoidosis you have a non-caseating granuloma, and you also have some bodies called asteroid bodies. Then in leprosy, again you have a non-caseating granuloma. Then there is something called cat scratch disease. That is going to give you a stellate granuloma. Then you have something called Dirk's granuloma in malarial infections. Those are the named types of granuloma. So here in this question, granuloma is a pathological feature of all except microscopic polyangiitis. Next question. Necrotizing epithelial granulomas are seen in all except, we already discussed this, the answer is leprosy. Here again they have given examples of granulomatous diseases and the type of granuloma seen in each. Now moving on to next question. Before that we'll take a short break. Okay, so moving on to the next question. Wound contraction is mediated by. So now we have come to the repair part. Tissue repair. So you have two words. You have repair and you have healing. Both essentially mean the same, but the difference is that healing is used when it is only the surface, that is if it is only the surface epithelia that is involved. But when the parenchyma is involved, the word used is repair. Essentially what is happening is, like in case there is a defect in the wall, you are putting cement. Similar to that, repair is going to happen. So that tissue repair can be by primary indention or by secondary indention. Now, primary indention, that is in case of mild superficial injury. Secondary indention is in case of deeper injury in which more amount of tissue and more depth has damage has occurred to more of the depth. So, here what is happening is, we'll talk about healing by first indention. So what is happening is, within 24 hours, you have neutrophils that are coming to the site. Then by day 3, please write this down with me, this is important. By day 3, you have macrophages coming. Then by day 5, neovascularization has taken place. Then in second week, you have proliferation of myofibroblasts and deposition of collagen. Now this collagen is initially type 3 collagen, then it is later replaced by type 1 collagen. Okay, so second week you have proliferation of myofibroblast and deposition of collagen. In third week, this proliferation is continuing and you have maximum collagen. Then after that, by 28 days, scar formation has taken place. Now, one thing you need to know is, in the first week, the wound will have 10% strength. By third month, please note it is third month, not week. There will be 70 to 80% wound strength. So, and this is the maximum of strength that can happen. So essentially what I mean is that, suppose you have a cut in your hand, scar is forming, whatever is there after third month, that will have about 70 to 80% of the strength of the original tissue, and that is the maximum strength it can attain. Okay. So moving on to the next question. Maximum collagen in wound healing is seen at, we have already discussed, third week. Okay, so which of the following is not a pyrogenic cytokine? So pyrogenic cytokine, what is the meaning of pyrogenic? Fever inducing. Where else have we heard about pyrogenics? And also pyroptosis. So as I said earlier, you have interleukin 1, which is the main leukin that is involved. Then you have interferon alpha, you have your TNF, and you have your interleukin 18. You have your interferon alpha. We'll take a short technical break right now. Okay, we had a short, small issue. Now I hope everything is clear. So, which of the following is not a pyrogenic cytokine? So, pyrogenic cytokines are interleukin 1. Then you have your tumor necrosis factor, that is your TNF, and you have your interferon alpha. Interleukin 18 is not a pyrogen. Okay. Next question. All are true about blood coagulation except. Okay. So, here you have the clotting pathway, that is the clotting cascade. You have the intrinsic pathway and the extrinsic pathway. So here, what is happening? In intrinsic pathway, negatively charged substances like glass beads. Now this is happening in laboratory, not in vivo. So this is what is happening inside our human body, but this is happening in the laboratory. So in the laboratory, what is happening is negatively charged particles. These activate factor 12. After factor 12, you have factor 11, then you have factor 9, after that you have factor 8, and then you have factor 10. So, what is the question? All are true about coagulation cascade except factor 10 is a path of both intrinsic and extrinsic pathway, that is correct. Extrinsic pathway is activated by, no, it is the intrinsic pathway that is activated by contact of plasma with negatively charged particles. Calcium is very important for coagulation, that is true. Intrinsic pathway can be activated in vitro, that is also true. So, here you have primary hemostasis is by platelet aggregation. Secondary hemostasis is by platelet, that is the fibrin clump formation. So, here you have, this is the site of injury. The first thing that is going to take place is all these platelets are going to come to the site of injury as a direct result of arteriolar vasoconstriction. That is leading to primary hemostasis. So here, in primary hemostasis, what is happening is, you have these platelet plugs that are coming here, and then these platelets plug, it is getting activated. First you have adhesions, then the shape changes. The platelets are circular, it becomes more that is plain. Then these granules are, some granules are released that result in recruitment of more platelets and in hemostasis plug formation. Now this much is what we call primary hemostasis. As a result of primary hemostasis, you have a platelet plug formation. Now, next what is happening is secondary hemostasis. There what is happening is you have deposition of fibrin. I hope that is clear. So, here in deposition of fibrin, you have tissue factor, then this phospholipid that is there, thrombin gets activated and fibrin deposition occurs. So, after some time, here there is repair that is happening, and as a result of that, after repair, the clot that is formed has to be resorbed. Then that is taking place. So here there are two things I have to tell you. First, if this is the endothelium, here you have factors, proteins called the von Willebrand factors. So, platelet comes and attaches there. So platelet attaches to the von Willebrand that thing through GP1B protein. Similarly, you have some other protein here wherein platelets get attached to each other. That particular protein is called your GP2B3A protein. So deficiency of this causes what is called your Glanzmann thrombocythemia, and deficiency of this factor causes your Bernard-Sallier syndrome. Okay, that is all I have to say about that question. We'll move to the next question. Vitamin K is responsible for carboxylation of which amino acid? Okay, so you think about vitamin K, you directly have to think 2, 7, 9, 10. Just think vitamin K, 2, 7, 9, 10. So these are the clotting factors that require vitamin K, and these are the clotting factors, and then you have two proteins like protein C and protein S that inhibit clotting. It is also vitamin K dependent, and whatever is vitamin K dependent, it is responsible for carboxylation of your glutamate. So vitamin K, 2, 7, 9, 10. Glutamate is the it is responsible for carboxylation of glutamate. So the factors are 2, 7, 9, 10. In addition, you have protein C and protein S. I hope that much is clear. Again, one extra point I have to say is warfarin is actually acting on this and causing bleeding. Now moving on to the next question. Which of the following is a procoagulation protein? Procoagulation protein is thrombin. These other proteins are anticoagulation. So we'll move on to the next question. Cause of edema is. Okay, so here you have a blood vessel. Now you have something called hydrostatic pressure that is around 32 here and around 12 there. So around 20. So hydrostatic pressure means that the difference in pressure between two ends, that is hydrostatic pressure. And then you have something called colloid osmotic pressure. This colloid osmotic pressure is the pressure that is applied by the proteins that are seen in the blood vessels to the wall. Okay, so if there is any, in normal homeostasis, you have a very delicate balance between the hydrostatic pressure and the colloid osmotic pressure. Now, when there is slight change in either of these, that results in edema. Now, edema can be of two types. If there is an increase in vascular permeability, what is going to happen? Whatever proteins that are here is going to come out of the cell. In other words, there is a protein rich exudate that is formed. Okay, in second case, suppose there is an increase in hydrostatic pressure, you have something called a transudate. So here what is happening is, there is fluid, but this fluid, that much protein and all are not going out. It is simply the fluid that is gone out. So that is transudate. So here Here we have some causes of edema. First, we will see the left-hand side. You have heart failure. Because of heart failure, you have capillary hydrostatic pressure that is decreased. Then because there may be decreased renal blood flow. Then because of that, because there is decreased renal blood flow, the renin-angiotensin, the RAS system, the renin-angiotensin system gets activated. As a result of that, there is an increase in sodium and water retention, and that causes an increase in blood volume, resulting in edema. That is one way in which edema is formed. Now, moving on to the right-hand side, you have malnutrition. Then you have hepatic stenosis, nephrotic syndrome. Essentially, what is happening there is a change in the colloid osmotic pressure. That is the protein. There is some sort of protein deficiency. So the colloid osmotic pressure is getting reduced. As a result of that, you have edema. So that is the two types in which edema can occur. So here what is from these: decreased plasma protein, increased lymph flow, increased ECF volume, increased plasma protein concentration. The cause of edema is only decrease in plasma protein concentration. Now, if there is increased lymph flow, suppose there is an infection like filariasis, for instance. In filariasis, what is happening? You have these filarial worms that are coming and blocking all the lymphatic channels. So as a result of that, what is happening? There is a decrease in, the fluid cannot go out. As a result, there is hydrostatic pressure, and as a result, it causes edema. But if there is increase in that lymph flow, it does not cause edema. It is only decrease that causes edema. Similarly, it is a decrease in the extracellular volume that causes edema, not increase. Okay, here we have different causes of edema. First one being increased hydrostatic pressure. Second one being reduced plasma osmotic pressure. Then lymphatic obstruction, sodium retention, and inflammation. Okay, we'll move on to the next question. 54-year-old chronic is brought by his son. He has developed increasing abdominal distension. So there is edema. Past 3 months, a physician aspirates abdominal fluid, straw colored, clear, found to have protein content, mainly albumin 2.3. Which of the following is the major? Albumin is 2.3% of albumin is present inside the fluid that has been aspirated. So from that, you can understand that there is some sort of exudative edema that is taking place. And the cause for that is decreased oncotic pressure. Okay. Now that we are on the topic of edema, there is something else about edema that I have to tell you. Now you have two words that you commonly come across in your day-to-day practice. You have hyperemia and you have congestion. Now, what is the difference between the two and what is the similarity between the two? So essentially, in both hyperemia and in congestion, there is increase in blood. That is what you need to know. Now, hyperemia is an active process. Congestion is a passive process. Hyperemia is because of increased arteriolar dilatation causing increased blood flow. On the other hand, congestion is because of venous stasis. That is, blood is not going back to the heart. So it is staying wherever it is present. So that is the difference between hyperemia and congestion. So as a result of heart failure or as a result of some sort of inflammation or obstruction, congestion can occur. Now this here, you can see the figure. This is what is called a nutmeg liver. Why I am talking about hyperemia and congestion here is that there are few interesting MCQ questions that are related to these. So first point being, In chronic venous congestion, that is CVC of liver, you have a characteristic appearance, that is nutmeg appearance. Okay, then in CVC spleen, you have something called a gamma Gandhi bodies. Then in CVC of lung, you have characteristic finding called hemosiderin laden macrophages. So again, hyperemia and congestion, essentially what is happening is you are having increased blood flow in both conditions, but pathogenesis of both is different. In hyperemia, it is an acute process which is caused by arteriolar dilatation, whereas congestion is a passive process which is caused by venous stasis. 3 points you need to remember: CVC liver, nutmeg appearance; CVC spleen, gamma Gandhi body; then CVC lung, you have hemosiderin laden macrophages. Now let's take a look at the next question. Extrinsic pathway of clotting factors is measured by prothrombin time. So prothrombin time, what is the normal prothrombin time? Around 14 to 18. So it measures the extrinsic pathway. This APTT, that is your activated prothrombin time, it measures your intrinsic pathway. Then your bleeding time measures both, and also bleeding time measures your platelet activity. Okay, then there is another thing called TEG, that is thromboelastography. So essentially it is an investigation which actually measures the coagulation of the coagulating ability of blood, and it has both APT, measures both intrinsic and extrinsic factors, and this you just need to know this word, that is TEG, and it is somewhat shaped like this. The normal TEG is shaped like this. That is all you need to know. Next, pale infarction seen in all except. I think we have already discussed that. So, infarct. What is infarct? Infarct is ischemia, that is area of necrosis caused by ischemia. Ischemia, we've learned, is loss of blood supply to a particular area. Now, your normal infarcts are wedge shaped, and you have two types of infarct, that is you have your red infarct and your white infarct. Red infarct is in areas where you have dual blood supply. For example, you have your lung, your GIT. White infarct is when you have end organ blood supply, and that is seen in your kidney, your liver, your spleen. So here, if you can see, this is the classic red infarct, that is the lung, and here in the kidney, sorry, this is not the kidney, this is the spleen. So you have pale infarct in the spleen. So here, remote kidney infarct now replaced by hydropic. Okay, so once, as I said earlier, once there is dual supply, if one area of blood supply is lost, some other blood supply can take place. But in areas where there is end organ blood supply, that cannot take place. So as a result of this chronic infarct, initially you will have a white infarct and later it is going to be replaced by what is called a scar, a fibrotic scar. Okay, let's move on to the next question. Hypercoagulability is seen in all except. So here there is a table directly taken from Robbins. So here we are moving on to the next topic, that is thrombosis. So you hear the word thrombosis, the first thing that should come to your mind is your virtuous triad. So virtuous triad, you have, it is a triad. So you have three factors. The first thing is endothelial injury. Then your second thing is abnormal blood flow. Then your third factor is hypercoagulability. So initially you will have endothelial injury. So thrombosis is an interplay of three factors. The first being endothelial injury. Now what can be the cause of endothelial injury? It may be several causes. For example, you can have atherosclerosis, then you can have inflammation, then you can have burns, then you can have microbial infections. All of that causes endothelial injury. Then abnormal blood flow. For example, abnormal blood flow in conditions like atrial fibrillation. Then when there is bed rest, that is venous stasis occurs, you can have abnormal blood flow. Now hypercoagulability can be divided into two. You can have congenital or primary hypercoagulability and you can have secondary hypercoagulability. So here you have both primary and secondary causes. So the most common primary cause of hypercoagulability is factor V mutation. Now factor V is also called lead-in factor. So lead-in factor mutation is the primary cause of primary hypercoagulability. Along with that you have increased levels of 8, 9, 10 or fibrinogen. Now secondary causes, as I said earlier, it can be prolonged bed rest, then myocardial infarction, atrial fibrillation, anything like that. So now we know which are the factors that cause a release in thrombus. So what is going to happen next? You have something called a thrombus. Now a thrombus can either propagate. So you have a blood vessel. Inside the blood vessel you have a thrombus. Essentially what is a thrombus? You have this whole thing where you have platelets, you have fibrin, you have plasma factors and everything that is causing an obstruction, and that is called a thrombus. So thrombus can propagate, then it can embolize, then it can dissolve, or it can recanalize. So these are the four options that a thrombus have. So a few points again. Any thrombus in CVS, that is in the cardiovascular system. Where in the cardiovascular system can you have thrombus? You can have something called mural thrombi, that is inside the lung. We are not dealing with that right now. You can have either an arterial thrombus or you have a venous thrombus. Whatever, whether it be arterial or venous, thrombus always propagates in direction of the heart. In direction of heart. So arterial thrombus is always going to propagate retrogradely, whereas venous thrombus will propagate forward. That is in the direction of blood flow. Okay. Now Some other interesting points. You have ante-mortem clot and post-mortem clot. So suppose you get a specimen for biopsy, you are doing autopsy on a body, then you find that in the muscle, suppose there is a blood clot that is there in a blood vessel in the muscle. So you have to think, was this blood clot formed before the patient died or was it formed after the patient died? Now, what is the significance? If the blood clot is formed after the patient dies, you don't have to think about it because the patient is already dead, and that thrombus itself could not have caused any problem to the patient. But suppose the clot is ante-mortem, you can say that particular clot, wherever in the vasculature it may be, will have a hand in causing the death of the patient. So you need to know whether it is ante-mortem or post-mortem. So for that, the first thing is, there are things called lines of ZAHN, ZAN. So in ante-mortem clots, you will have lines of ZAN. Post-mortem clot, no lines of ZAN. So what are these lines of ZAN? Here you will have some pale area, then you will have red area, then pale area and red area, pale red, pale red. Now this pale red, it is because of this red areas is formed by RBCs and this pale area is formed by rest of the exudates. Okay, that is all about thrombus, I think. Another thing you have to know is, there is something called a Trousseau syndrome. That is migratory thrombophlebitis. Now it is usually seen in disseminated cancer. So essentially what is happening is, you have a thrombus that is migrating from one place to another, and it is usually a result of disseminated cancer. So as we said earlier, thrombus can either propagate, which we talked about, that is, if it is an arterial thrombus, it propagates retrogradely, whereas if it is a venous thrombus, it propagates in the direction of blood flow. In both cases, it propagates in the direction of heart. It can dissolve, it can recanalize, or it can embolize. So what is meant by embolism? Now embolism is a detached intravascular solid, liquid or gaseous mass that is carried from one place to another by blood. So what are the most common important examples of embolism that you can think of? So first thing is amniotic fluid embolism. Though not very common, it is a very almost sure cause of maternal mortality, amniotic fluid embolism, and it can occur in both during normal delivery and during cesarean section. So that is one thing. Then second thing, have you heard of something called fat embolism? Now fat embolism, the clinical scenario is going to be a male or a female in young age who is bedridden as a result of femur fracture, long bone fracture of the leg. So one fine day, the patient will develop breathlessness, pulmonary edema, pulmonary failure, and will die. So that causes fat embolism. Then you have something called a saddle emboli. What is your saddle emboli? You have your pulmonary artery and your pulmonary vein, and that is actually present like this. That is, it saddles the aorta. So if there is an embolism somewhere there, it is called a saddle embolus. That is all about embolism. So here you have, okay, our first question was congenital hypocoagulability seen in all except, the answer is antiphospholipid syndrome. Lines of Zan are found in, the answer is thrombus. In particular, the answer is anti-mortem thrombus. Then you have chicken fat clot is seen in, okay, the typical appearance of post-mortem thrombus has been described as chicken fat appearance, because it is almost yellowish in color. Here you have a picture, both chicken fat clot and currant jelly clot, these are both post-mortem clots. And here you have an anti-mortem clot along with your lines of Zan. Okay. All the following are true about DIC except. Okay, so DIC, what does DIC stand for? It stands for disseminated intravascular coagulation. So here, essentially, what do you mean by disseminated intravascular coagulation? It is the most common patient that you will see is somewhere, maybe a pregnant lady with eclampsia or preeclampsia, or it may even be a patient in sepsis where you can get DIC. So what is the pathogenesis that is happening? So here, either because of endothelial injury, which may be because of burns, then it may be because of microbial toxin, it may be because of mechanical trauma, whatever the reason may be, you will have massive tissue destruction, sepsis, endothelial injury. As a result of that, you will have tissue factor. Then this tissue factor release will cause thrombosis everywhere. And this, as a result of this thrombosis, there is thrombosis. What is thrombosis? Thrombosis means clot formation, right? So clot is forming everywhere throughout the vasculature. As a result of that, all the clotting factors and all the proteins, these get taken up. So as a result of that, bleeding occurs because there is no clotting factors left to control the bleeding, because all the clotting factors have been taken up by this disseminated thrombus. That is one way of pathogenesis. Then, as a result of this widespread microvascular thrombus, you can have fibrin formation, fibrinolysis. As a result, you can have bleeding. Then you have clotting factor lysis. Again, you can have bleeding. Then it can cause vascular occlusion, again causing tissue damage. So the question here was, what are true about DIC? So consider each thing. So platelet count. So how is the platelet count going to factor in DIC? As I said earlier, you are going to have thrombus everywhere. So as a result of that, your platelet count is going to decrease. Then you have things like your FTP and your D-dimer. What is FTP? FTP is your fibrin degradation protein. One of such protein is your D-dimer. This is all going to be increased because all the clots that are there, it is continuously being broken down by the fibrinolytic system. Then you have fibrinogen. Fibrinogen, what is fibrinogen? Fibrinogen is the thing from which fibrin, that is the clot formation, occurs. So fibrinogen is also going to be taken up. So fibrinogen level is going to decrease. Then you have PT and you have APTT. What does PT? PT measures the extrinsic pathway and PT measures the intrinsic pathway and APTT measures the extrinsic pathway, and both are going to be increased. So that is all about that question. Going on to the next question, the appropriate number of genes contained in human genome is 30,000. So this is just, I just included this question because I think it's a very interesting fact. So Duchenne muscular dystrophy is, okay, so now we have reached another topic, that is genetics. So, first of all, what do you mean by genetics? Study of genetic material. So, what genetic material? Some very small, very simple things. You have two types of bases. You have purines and you have your pyrimidines. Your purines are adenine, guanine. Your pyrimidines are cytosine, uracil, thymine. DNA has adenosine, guanine. A always pairs with T. Guanine always pairs with cytosine. If it is RNA, instead of T, you have uracil. That is the very basics of genetic material. Now, what do you mean by a mutation? Webster's dictionary describes mutation as a sudden, drastic, inheritable change in the genetic makeup of an individual. So, as far as we are concerned, mutation can occur by three different ways. Either there can be a mutation in a single gene, or there can be some chromosomal defect, or there may be something called complex multigenic disorders. Okay, so we will see each in short. Mutation in a single gene. You all might have heard of the word Mendel. Not mental with a T, Mendel with a D. Mendelian, you all must have this, you have seen this type of square where you have a TT here and a TT here, and it can have many, it can have four options like that. So whenever there is a single gene defect bar mutation, that is called a Mendelian disorder. Okay. And we call it Mendelian disorder because there is a definite way in which this mutation is transmitted. Okay. So, before we begin, a few words actually. Penetrance. What do you mean by penetrance? Penetrance of a gene means that if there is a mutation in that gene, there will be a disease. That is high penetrance. So penetrance is whatever change that is there in the genetic material, how much it translates phenotypically. That is how much it translates into practice. That is called penetrance. So suppose there is a mutation and that mutation causes a disease. You can say that mutation, whatever it is of high penetrance. Suppose there is a mutation but the disease is not caused, that is, or the disease is caused in very low amount. There you can say it is low penetrance. I hope that is clear. So in all sorts of Mendelian disorders, you have high penetrance, and in other disorders That is, for example, polygenic. What do you mean by polygenic? Polygenic means that many genes can cause a disease. That is, one gene can have many alleles and many mutations, and that will cause a disease. That is called your polygenic type of disorder. That will have a low penetrance, because just because there is a presence of a mutation does not guarantee that there is a disease. So mutation is not equal to disease. In that case, we say it is low penetrance. Okay, so first type of thing we'll talk about, point mutation. So you have your DNA, you have some coding sequences and some non-coding sequences. Coding sequences ultimately results in protein, then non-coding sequences does not result in protein. That and all we'll learn in biochemistry. Point mutation within a coding sequence. What is going to happen? First of all, what do you mean by point mutation? A single base pair change happens. For example, A changes from A to G, or A changes from A to C. What is going to happen? There is a single base pair that is changed. It can have two things. That is, either it can be a missense mutation, or it can be a nonsense mutation. So missense mutation, in missense mutation what is happening is you have, instead of one amino acid, instead of one base in that coding sequence, some other base is coming. For example, T is coming instead of A. It can have two types, it can have two outcomes. Either it can be conservative, wherein even though it is changed, the same protein or the same amino acid gets translated. Okay, so is there a mutation? Yes, there is a mutation. It is a missense mutation. Missense means there is a single change from one amino acid, from one base, purine or pyrimidine base, to the other. And in that it can have two outcomes. In one outcome, even though there is a change in base pair, the amino acid that is the result remains the same. In that case, you call it conservative missense mutation. Now, suppose there is a different amino acid that is formed. That is a non-conservative amino acid, non-conservative missense mutation. Again, point mutation within a single base pair. You can either have missense mutation or nonsense mutation. In missense, what is happening is a single purine or pyrimidine base is changed. For example, A is changed to T, or C is changed to D, whatever it may be. It can have two outcomes. In one outcome, even though there is a change, it is the original amino acid itself that is getting translated. So that is your conservative missense mutation. In non-conservative missense mutation, what is happening is, because there is a change in a base pair, there is a change in the amino acid that is translated. So that is your non-conservative mutation. For example, we have all learned, in the sixth beta globin chain somewhere, glutamic acid changes to valine, and that causes what is called your sickle cell anemia, right? That is an example of a missense mutation. Now, nonsense mutation, what is happening is, there is a change in a single base pair, but instead of another coding or another codon that is there, it is a stop codon. So as a result, the translation gets stopped. That is called a nonsense mutation. For example, you have CAG, that is a codon which translates for glutamate. Now, instead of that C, if it U comes, if it is UAG, then that translation is stopped. It is a stop codon. And the example for such a condition is your thalassemia. In specifically your B0 thalassemia. Okay. Now that is point mutation that is happening in a coding pair. Suppose this point mutation happens in a non-coding sequence. So this non-coding sequence, these have an important role in our genetic makeup. These are your promoter and your enhancer regions, right? So if there is a mutation there will be lack of transcription factors. That can also cause a disease. Now your third type, you can have deletions or insertions. Suppose you have something like TTA, TTG, TTC. This is your coding sequence. Suppose if the deletion is happening in multiples of 3, in 3 or multiples of 3, there is just the reading frame, that is this reading frame, that is the translation will take place. The only difference is that sometimes a different amino acid gets bound or there will be a deletion in that gene. Suppose if the number of base pairs that are getting deleted or inverted is not in multiples of 3. So what has happened? So what happens? So you have TAA, you have TTG. Here just G is getting deleted. So what do you have? You have TT. So as a result, what is happening? Here next you have TGA, TCA, like that. But here instead, because one is deleted, the frame of the entire the transcription frame shifts entirely. Here, I will explain again. Suppose you have your mutation here. This is your coding sequence. You have a TTG here. Now once this TTG gets deleted out, what is happening to the protein? In the protein, what is happening is that the frame of that transcription remains the same. There is either another protein or another base pair that is coded instead, or there is something that is not coded. That is normally what is happening. Now suppose this deletion happens where the number of deletions that is happening is not a multiple of 3. So here this G is deleted. That G alone is deleted. So here after TTA, TTG, it is TTT. That is, this T gets read with this other 2 T's. So there is something called a frame shift mutation. I hope that is clear. Then suppose we talked about mutations, that is point mutations in coding sequence, non-coding sequence, in deletion sequence. Then suppose some trinucleotide repeat there is some sort of error in the trinucleotide repeat sequences. For example, you have CGG, which codes for familial mental retardation gene. That is FMR1. That causes your Fragile X syndrome. Okay. Now, one thing I have to say about your trinucleotide repeat sequence, it is always it is just your C and G base pairs that are involved. Okay. So, what did we learn right now? We learned what all happens in Mendelian disorders. Mendelian disorders means disorders in which a single gene has some problem, has some mutation. Initially, if there is a point mutation in a coding sequence, you can either have a missense mutation or a nonsense mutation. In missense mutation, you can have two types. That is, you can have conservative and non-conservative. In conservative, what is happening is that even though the base pair is changed, the amino acid, the result remains the same. That is conservative. In non-conservative, base pair is changed, the amino acid is also changed. Now, coming to nonsense mutation, a base pair is changed. Because of that change in base pair, stop codon gets translated. So what happens? There is stopping of that translation. Now, secondly, if there is a mutation in the non-coding sequence, you can have some sort of transcriptional defects. Thirdly, if there is a deletion or inversion, which is in a multiple of 3, that means there will be some change in the protein that is formed, but the frame of the reading of the codon remains the same. But in deletion or inversion is occurring not as a multiple of 3, what you get is you will have a frame shift mutation. Now, another word is trinucleotide repeats. So, trinucleotide repeats, two things you have to remember. First thing is, it is always C and G that is involved. Cytosine and guanosine are the only bases that are involved in case of trinucleotide defects. Second thing is CGG, it results in familial mental retardation gene, that is FMR1, and results in Fragile X syndrome. Okay. Now, again, few points which are important for your entrance. In Mendelian disorders, classically you can have either you can have a dominant allele, a recessive allele. Another person can have a dominant allele or a recessive allele. In some cases, there is something called a codependence. Codominance, not codependence, codominance. Now that codominance, the example is HLA grouping and your normal blood grouping. Okay. Then again, two words. First word is pleiotropism, pleomorphism. What do you mean by pleomorphism? A single mutant gene has many different end effects. A single mutant gene having many different end effects, that is called pleomorphism. On the other hand, you have something called a genetic heterogeneity, wherein mutations at several genetic loci can lead to same disease. For example, your childhood deafness. Okay. Again, pleiotropism, mutation in a single gene that is resulting in many side effects. That is called pleiotropism. Genetic heterozygosity, mutations at several genetic loci can cause same effect. For example, childhood deafness. Okay. Now, we were talking about all of this because the next thing you have to learn here is Duchenne muscular dystrophy. Okay, just Duchenne muscular dystrophy is an X-linked disorder. So, I have a mnemonic for this X-linked recessive disease. It is not my own mnemonic. It is what I found on the internet. I can't take credit. I will tell you what it is. It is oblivious female will give her boys her X-linked disorder. So, O, oblivious. O stands for ocular albinism. Female, F stands for Fabry's disease. Will, W stands for Wiskott-Aldrich syndrome. Then G stands for G6PD. H stands for Hunter syndrome. B stands for Bruton's A gamma globulinemia. Then H stands for hemophilia. Then X-linked, L stands for Leishman syndrome. And D stands for Duchenne muscular dystrophy. Okay. In that similar vein, you have X-linked dominant disorders, wherein the mnemonic is again taken from net. I can't take credit. Red ribbon for all children. So here R stands for rickets, that is vitamin D dependent rickets. Then other R stands for Rett syndrome. Then F stands for Fragile X syndrome. Then A stands for Alport syndrome. Then C stands for Charcot-Marie tooth disease. Okay. So Duchenne muscular dystrophy is an X-linked recessive disorder. So in Duchenne muscular dystrophy, what is happening? It is an X-linked recessive disorder. So you have the X chromosome in which you have a gene called dystrophin. Now, if there is complete absence of that gene, you have your Duchenne muscular dystrophy. It can be because of deletion, it can be because of frame shift mutation. Now, suppose there is abnormal dystrophin gene, then you will have what is called abnormal dystrophin gene, you will have Becker's syndrome. So that is all about that question. We'll take a small break now. So we'll continue. We were talking about Duchenne muscular dystrophy, which is an X-linked recessive disorder. So we'll move on to the next question. So here, this is just a histological picture wherein you see atrophy of muscles. This is how Duchenne muscular dystrophy or muscular dystrophies or any type looks like. Now, you have vascular type of EDS, that is Ehlers-Danlos syndrome. So here, your Ehlers-Danlos syndrome, what is the basic defect is that there is a mutation in collagen. Okay. Now, because of a mutation in collagen, it can have several defects in several aspects of the body. So, because of that, because it affects bone, you can have something called osteogenesis imperfecta. Then it can cause Alport syndrome. Then in skin, it can cause epidermolysis bullosa and so many things. Now, talking about the type of inheritance in EDS, you have type 4 and type 7, which are autosomal recessive. All others are autosomal dominant. Now, what are the clinical features? Now, here in this session, I am talking about just a few disorders because these are the frequently asked disorders, and that is why you need to focus on these disorders. So, speaking about the clinical features, you can have hyperflexibility of joints, then you can have hyperelasticity of skin, then because of that you can have fragile skin, a fragile lens and bruises and all of that. Now here the question is vascular type and it is type 4. The problem with vascular EDS is that there may be rupture of coronaries of your colon or visceral vessels. So rupture of vasculature can occur in EDS type 4. Here you have a picture that shows the hyperextensibility of skin. So moving on to the next question, you have Marfan syndrome, there is defect in protein fibrillin. So Marfan syndrome, the classic definition is you will have a tall stature, long fingers, then you will have bilateral subluxation of lens, then you can have mitral valve prolapse, then you can have abdominal aneurysm, then you can have aortic dissection and everything. So the incidence of Marfan is 1 in 5,000 and the gene involved is FBN1 gene. So here as you can see, it is in chromosome 15. You have, in a healthy person, you have 2 FB1 genes, normal fibrin, strong protein. In a patient with Marfan syndrome, you will have some problem in the chromosome 15, altered or too little fibrillin, fibers are too weak. That is all about Marfan syndrome. Now, next question, hemophilia is associated with X chromosome. This we have already dealt with, and here again I gave you a mnemonic for X-linked disorders. I think I have not given you a mnemonic yet for autosomal disorders. So, okay. So, disorders, you have 4 types, as I mentioned earlier, autosomal recessive, autosomal dominant, X-linked recessive, X-linked dominant. I think we have already seen X-linked recessive and X-linked dominant. So now autosomal dominant. Here, again a mnemonic that I've taken from the net. Very powerful dominant human. So, V in very stands for von Willebrand disease, or your VHL, that is von Hippel-Lindau syndrome. Then you have pseudo hypoparathyroidism. Then that is P. Then dominant, each letter has a disease. So D you have myotonic dystrophy, that is dystrophica myotonia, that is myotonic dystrophy. Then O you have osteogenesis imperfecta. Then Osler, please look in that, Osler-Weber, I think, syndrome. Then you have M, you have Marfan syndrome. Then you have I, that is intermittent porphyria. Then you have N for neurofibromatosis. Then you have A for achondroplasia. And also adult polycystic disease kidney. Then N for Noonan syndrome. Then T for tuberous sclerosis. Then you have dominant human H. H for hereditary spherocytosis. Then you also have other H diseases like you have your Huntington's chorea. Then you have your HNPCC, that is hereditary polyposis syndrome. Now that is all about autosomal dominant diseases. Now let's come to the next type of diseases, that is your autosomal recessive. Again, mnemonic is A, B, C, D, E, F, G, H, I. So A stands for albinism, alkaptonuria, ataxia telangiectasia. B stands for beta thalassemia and your sickle cell anemia. Then C stands for cystic fibrosis, congenital adrenal hyperplasia. See, D stands for deafness. E stands for emphysema. F stands for Friedreich ataxia. G stands for Gaucher's disease. H stands for homocystinuria. And I stands for inborn errors of metabolism. Okay. Now, from this topic, from genetics, some sort of questions that you can expect is, what is the type of inheritance you see in so and so, that is in Duchenne muscular dystrophy, in Becker's dystrophy, whatever that is one type of question. Then the second type of question is you will have some pedigree charts that are given. So in dominant disorders, one of the parent is always going to be affected. Okay, so whenever you see a pedigree in which there is always a parent that is affected, the child is also going to be affected, you can surmise that it is a dominant kind of disorder. Parent is always affected. Now, if it is a recessive disease, parent may or may not be affected. Not required that the parent is affected. The parent can even be a carrier. So here, suppose A is the disease, A is the dominant disease thing, and this A is normal. If this alone causes the disease, you can say it is a dominant disease. If you need two alleles which are affected, you have to say it is a recessive disease. Okay, so when you get a pedigree, first thing you check is it going to be X-linked or not? How to check if it is X-linked or not? If it is X-linked, most likely it is the males that are affected. Okay, so you see a pedigree chart, and in that, first think if it is the males that are affected, if it is only the males that are affected, only in few cases will females be affected. So in that condition, you can say it is X-linked. Now, second thing is, so first thing you have to check is whether it is X-linked or not. Second thing you need to check is whether it is dominant or not. If the parents are affected and the child is affected, it is dominant. If the parents may or may not be affected, and if the child is not affected, it may be recessive. Okay. So moving on to the next question, which of the following is an autosomal dominant disorder? The answer is Huntington's chorea. Which of the following is not an X-linked condition? So here this is the answer, facioscapulohumeral muscular dystrophy. It is an autosomal recessive condition. Autosomal dominant condition. Now Duchenne muscular dystrophy is X-linked. Okay. Now, which of the following is an example of non-Mendelian inheritance? Okay, so here you have a few words. The first one is genomic imprinting. What do you mean by genomic imprinting? Genomic imprinting. So, any disease, whatever the disease may be, it will have two alleles. Most usually, it has two alleles. That is, if these are the two alleles, one is paternal, one is maternal. However, in some condition, it is only one of that allele that has the upper hand. Even though two alleles are present in that patient, in that person, need not be a patient, it is only one allele from one particular parent that is active. Okay, so what I am saying is, the word imprinting means that either the paternal or the maternal allele has been inactivated. Okay, so first of all, when we began this topic on genetics, the first thing we learned is that there are some diseases which follow Mendelian characteristic. That is, there are two alleles, and two alleles, either one may be dominant or recessive or whatever, and it happens in different frequencies, and that is what results in the disease. Now, in some diseases, it does not follow this Mendel's laws. Those kind of diseases are called non-Mendelian disorders, and in that you have three terms. First one is genomic imprinting. So what is genomic imprinting is that you have, for every gene you have two alleles. One is paternal, one is maternal. Now in genomic imprinting, what is taking place is that either the paternal or the maternal gets inactivated. So even though there are two alleles in that patient, functionally there is only one allele. So genotypically there are two alleles, but functionally it is only one allele. That is your genomic imprinting. Now, how does this genomic imprinting take place? It is as a result of epigenetic changes. What do you mean by this epigenetic changes? Epigenetic changes is that after the gene gets translated, after the protein is formed, whatever is taking place on that gene, that is called epigenetic changes. A few examples are, you have DNA methylation, you have histone deacetylation, then you have methylation. Okay, now let me just add a page. Okay, you have two very common diseases that have something to do with this genomic imprinting. That is called, you have Prader-Willi syndrome and you have Angelman syndrome. So Prader-Willi is on chromosome 17 and Angelman is on chromosome 15. So Prader-Willi P, paternal allele gets inactivated. Okay, so in Prader-Willi syndrome, in chromosome 17, even though there are two alleles, one from the father and one from the mother, it is only the maternal allele that is active because the paternal allele has been inactivated. That is Prader-Willi syndrome. On the other hand, you have Angelman syndrome, which is the direct, I won't say direct opposite, which is opposite to this. It is present in chromosome 15 and here it is the maternal allele that has been inactivated. Okay. So one other thing is, there is a protein called UBE3A that has been associated with this Angelman syndrome. And another word for this Angelman syndrome is happy puppet syndrome. Now, both cases you will have intellectual disability and other disorders, but in Angelman, the patient, the child is going to be expressionless and it is called happy puppet syndrome. So, that is about genomic imprinting. And next you have another word there, that is uniparental disomy. So in uniparental disomy, what you mean is that in genomic imprinting, you have two alleles. both from different parents. But in uniparental disomy, you have two alleles, but both are from the same parent. That is, two alleles or two genes or whatever, both are from same parent. I hope that is clear. Now, mitochondrial inheritance, again from mother to son, mother to children. All of these are examples of non-Mendelian disorders, and here you can see a table where you have examples of conditions of each. Okay. Next question. The phenomenon where subsequent generations have the risk of earlier and more severe disease is known as anticipation. Okay, so essentially what it means is that if I have a disease, I have a disease in a severity of say 6 out of 10. In my next generation, the severity will be 8 out of 10. And after that, in the generation after that, it is going to be 10 out of 10. So in subsequent generation, the defect gets more and more severe. That is called anticipation. The next question, long arm and short arm of a chromosome are called Q and P. Okay, so here, how to remember? P stands for petite. Petite means small. Okay, so P stands for petite. Petite means small. So petite is always small. So that is the short arm. Q happens after P. So it is the long arm. Clear. Now go next question. Here you have the P arm and you have the Q arm and the P arm, and you have the centromere that is there. And in relation to the centromere position, you have metacentric, that is, it is almost in central. You have submetacentric, acrocentric, almost at the end. And you have telocentric, where it is at the end. Okay. So, your next question. Which of the following is used for separation and detection of RNA? Okay, so here again, you have several types of blots. So what is happening? In northern blot, it is RNA that is being amplified. In southern blot, it is DNA that is amplified. In western blot, it is proteins that are amplified. In eastern, it is post-transcriptional factors that are amplified. So how to remember this? Again, a very common thing. North, if this is your India, if this is India, you have, this is your south, this is your north, and this is west. Okay. North has, what do they eat? I mean, it is stereotyping in a sense, but it is just a way for you to remember easily. North is famous for its roti, so R. North, you have RNA. South is famous for dosha, so you have DNA. West is famous for pizza. Simply, it is protein. What is left is eastern. So here, what is important is that you will get some pictures, and one type of question is like this, like which is the technique used for separation of RNA, which is a technique used for separation of DNA, and questions like that. The other type of question that you get from this type of other type of question that you can get is, you will have something like this. So here you can see, without naming, suppose this naming is not there. You just look at this. What do you get? You see the word protein. So if you see the word protein, you have to think it is western blotting. Okay. Secondly, here you can see RNA. You see the word RNA somewhere in the beginning? It is southern blotting. Okay. Then here you have DNA. You see the word DNA, what is it? It is always, if you see the word DNA, it is always southern blotting. If you see the word RNA, it is always north, not south, my mistake. RNA is always northern blotting. What is left is eastern blotting. Now, at this juncture, I have to say a few things about these kind of things. So, PCR. What do you mean by PCR? Polymerase chain reaction. So, what is happening in your PCR? You have three processes. First, essentially PCR is a method by which DNA or whatever it may be is amplified so that you can use it to study further, to do further studies on that. So that is one thing you have to remember. PCR is polymerase chain reaction and the steps is DAE, that is denaturation, annealing and extension. I hope that's clear. Now we'll move on to the next question. Thalassemia occurs due to which of the following mutation? The answer is splicing. We have already explained. Now LE cell. Okay, what is LE cell? LE cell is a neutrophil that has engulfed another neutrophil. And you have some other cells called TART cells, which is a monocyte that has engulfed some other nuclear material. Okay. So, a false negative tuberculin reaction may be obtained in all of the following except children positively tested with tuberculin test. So, here you have conditions where you get a false negative Mando test and false positive Mando test. Now, if mother is donating her kidney to her son, this is an example of which of the following. Okay, so you have organ donation. Organ donation, you have four types. Isograft is from identical twin. Xenograft is from another species. Autograft is from self. That is, you take skin from your leg and you keep it on your scalp, or vice versa. It is called autograft. Then allograft. Allograft, it may be a relative, it may be your mother, it may be your partner, whatever it is. It just means that the donor is a person of the same species, unless it is your identical twin, in which case it is isograft. All other people, including your mother, your sibling, your father, whoever it may be, it is called allograft. So serum sicknesses. So here we are coming to the next topic, that is hypersensitivity. Hypersensitivity, very, very, very important and very in its most basic form. Type 1 hypersensitivity is IgE mediated. Type 2 hypersensitivity is antibody mediated. Type 3 hypersensitivity is immune complex mediated. Type 4 hypersensitivity is cell mediated and it is delayed. So, serum sickness is type 3. It is an immune complex. So, here you have the different types of hypersensitivity reaction we have learned. So here you have mechanism for each. Type 1, most common example is your anaphylaxis. Type 2, antibody mediated. So that is your immune hemolytic anemia, your good pasture syndrome. Type 3, that is immune complex mediated. That is your SLE. Then you have serum sickness. Then you have Arthur reaction. Type 4, you have contact dermatitis. Type 1, diabetes, etc. So here you have the pathogenesis of all. I'm not going to discuss this in detail. First one, you have type 1, that is IgE mediated. You have an allergen. Essentially, IgE gets activated and that IgE causes immediate hypersensitivity reaction. Other examples are bronchial asthma, allergic rhinitis and food allergies. Now second one is your type 2, that is antibody mediated, wherein this is the pathogenesis. All of these figures are important because these have all been taken from Robbins, and what is going to happen is they will give this figure. Some cell will be masked, some cell or some protein will be masked, and you will be asked to answer what that protein is. So please make sure that you go through this properly. Type 2 hypersensitivity, you have autoimmune hemolytic anemia, autoimmune thrombocytopenic purpura, you have pemphigus, you have vasculitis, rheumatic fever, myasthenia gravis. Now here type 3, that is immune complex, you have only a few examples. Post-streptococcal glomerulonephritis, PAN, that is polyarthritis nodosa, serum sickness, Arthur's reaction. Then you have your type 4, again rheumatoid arthritis, multiple sclerosis, type 1 hypersensitivity, IBD, psoriasis, contact sensitivity. Now we'll move on to the next question. A 23-year-old presented with jaundice, pallor, 2 months. Her peripheral smear shows presence of spirochetes. The most important investigation to arrive at the diagnosis is which of the following? So think, you have a 23-year-old female who is presenting with pallor. So what is going to be your primary diagnosis? A woman in reproductive age presenting with pallor, first thing you have to suspect is always iron deficiency anemia. Okay. But here there is a twist. The patient does not only have pallor, the patient also has jaundice. So what does that jaundice signify? The jaundice signifies that there is some breakdown of RBCs as well. Okay, so it is not just an iron deficiency. If it is iron deficiency, the peripheral smear picture you see is microcytic hypochromic anemia. But here you have something called spirochetes. Now spirocells, like you see the word spirocyte, schistocyte, then macroovalocyte, you see all these words, you have to think it is a type of hemolytic anemia. What do you mean by hemolytic anemia? Anemia can be of two types. It can be because of decreased production or increased degradation. If there is increased degradation, the patient is going to have, what is happening? RBCs are broken down. As a result of RBC breaking down, you have hemoglobin. Then hemoglobin, when it gets broken down, clinically, it is seen as serum bilirubin increased. That is already mentioned as jaundice. So it is a hemolytic anemia. And in that age group, we have to suspect something called immune hemolytic anemia. And for that, the test that you do is your Coombs test. It is a very favorite question of all examiners. You have Coombs test, which is of two types. You have direct Coombs test and indirect Coombs test. Here, as you can see, patient's blood, when patient's blood is incubated with antibodies, it is called direct Coombs test. Now, it is when patient's sera is with antibodies, it is indirect Coombs test. Now, this Coombs test is a very frequently asked question. So again, direct Coombs test, blood sample from a patient with immune hemolytic anemia, that is, blood sample is taken, first step. Now, the blood sample, the RBCs are washed. Why are we washing the RBCs? Is we don't want any unneeded antigens and antibodies when we are doing this test, which is why RBCs are washed. You just take it at face value. So, RBCs are washed, and then you have anti-human antibodies that are there. Now, if the patient's blood has antibodies against its own blood, the test will be positive. Okay. In indirect test, it is patient's blood that is taken. In indirect test, it is patient's serum that is taken. Now, patient's serum is taken and donor's blood is added. Now, once patient's serum and donor's blood is added, what is there is that the recipient's antibodies that target the donor's red blood cell form complex. Recipient's antibody that target the blood cells form complex. As a result, then again you do this, like you do it with anti-human antibodies. If you get a positive res, it means that the patient's sera also contains antibodies. Okay. For easy remembering, only two things. In direct, you are taking patient's blood. In indirect, you are taking patient's sera. In direct, you are directly adding anti-human antibodies. Here, first you are adding donor's blood and then you are adding anti-human antibodies. I hope that's clear. Now moving on to next question. Cold hemagglutinin is associated with. Okay, so we were talking about immune hemolytic anemia. So you have two things known as warm antibodies and cold antibodies. Now, warm antibodies, example, warm antibody means that in 37 degrees Celsius, agglutination is going to take place. Now, cold antibody, what it means is that when you heat, agglutination that has taken place will dissolve. Okay, so cold hemagglutinin is associated with Donat-Landsteiner antibody. Now, warm antibody type, you have primary and secondary. Most common is your SLE antibody. Cold antibody type, usually your mycoplasma, then your mononucleosis antibodies. Now, which of the following protein defects can cause hereditary spherocytosis? Except glycoporin A. So, what is hereditary spherocytosis? It is an autosomal dominant condition in which the peripheral smear is going to show so many spherocytes. Now, how do you identify a spherocyte? Spherocytes are identified by the fact that they have no central pallor. We are coming, okay. It is coming later on. For now, you just know that this is the RBC membrane and you have 3 types of protein. Band 3, ankyrin and 4.2. Band 4.2, band 3 and ankyrin. So, a defect in any of this can cause some membrane instability. As a result of that, it will result in the formation of spirochetes. That's all you need to know. And this is the blood picture. So, this you can see, these are not your normal RBCs. These are, normally we consider a size of normal RBC is similar to that of a lymphocyte. Okay, so any cell that is smaller than that RBC is called microcyte. So if this you can see, you have a small cell with no central pallor. No central pallor. So those kind of cells are spirochetes, and whenever you see a smear with so many spirochetes, the first diagnosis should be hereditary spirocytosis. And if you see some other cells in addition to spirochetes, and if you know the patient is not hereditary spirocyte, the second thing you have to suspect is hemolytic anemia. Now, one of the tests that we do for this hereditary spirocytosis is called osmotic fragility, which has absolutely no clinical value, only histological importance. So what you do is essentially you take patient's blood and you take blood from a control. Who is the control? Someone who is the same age and gender as the patient without any disease. A normal person of the same age and gender as the patient. So what you do is you prepare stock solutions of sodium chloride of varying concentration, and you put 1 or 2 drops of control blood and patient's blood in each of these test tubes. So as you can see, here this is the normal. This is normal patient, and hemolysis occurs in whatever concentration it may be. In a patient with hereditary spirocytosis, osmotic fragility is increased. As a result of that, you can see hemolysis starts at a lower concentration. And then you draw something called a S-shaped or a discoid graph. So if there is a shift to right, it is hereditary spirocytosis. If there is a shift to left, it is either sickle cell anemia or thalassemia. As I said earlier, it has absolutely no clinical significance. The only significance is of histological significance and the fact that this is usually asked for exams. Okay. Again, you have warm antibody and cold antibodies. We discussed this. The answer is SLE. So, warm antibody types. Warm antibody, another point I forgot earlier was warm antibody is always IgG, while cold antibody is always IgM. So, you have several types of antibody molecules, that is IgM, IgG, IgA, IgD, IgE. So IgM is the first one that is formed. Then IgG is the one that can cross the placenta. Then IgA is the one that is present in the mucosal surface. IgE is associated with allergy or anaphylaxis. So warm antibodies are always IgG type and it is active at 37 degrees Celsius. Cold antibodies are IgM type and it's active at 37 degrees Celsius. And here are the examples. So schistocytes are found in all of the above. So here, this is another table of all the cells that you see in a peripheral smear. You please go through this properly, because one of 2 years back, a NEET question was, you had a peripheral smear and you had so many cells there and you were asked to identify each cell. So RBCs, biconcave shape, approximately 8 micrometer, no nucleus, no metabolic machinery. Neutrophils, as I said earlier, in a peripheral smear, you can see 3 types of cells. You can see RBC, you can see WBC, you can see platelet. Platelet you won't be able to see because it is seen only as fragments, some purple color. And in peripheral smear, as I said earlier, no fixation is required. You can directly stain because the stain contains a fixative, and it is a Rowanowski type of stain. Most common examples are Leishman and Giemsa. Okay, so this is RBC. WBC can be granulocytes and it can be agranulocytes. Granulocytes example is neutrophil, eosinophil, basophil, agranulocytes, monocyte and lymphocyte. So RBC is biconcave shape, and neutrophil you have abundant granules, and all these granules may contain some factors called myeloperoxidase. Then it may have something called lysozyme, which is not of much importance. Eosinophils. Now, how to differentiate a neutrophil and a eosinophil? It does not depend on the number of lobes. Now, usually when we study in our 12th standard and all, we study it as bilobed cell is eosinophils and multilobed cell is neutrophil. But eosinophils can also be multilobed and neutrophils can also be bilobed. So what you have to do is, in case of eosinophil, you will have this reddish-orange granule. Reddish-orange granules. In case of neutrophil, you will have some purplish granule. That is the only difference. Now, basophil, again, basophil is not normally seen. In case basophils are seen, you have to in more than 2% in a W smear, you have to suspect some sort of problem is there with the myeloid lineage. Again, dense basophrenic granules you will see, that is basophil. This is wrong. Please don't mind. This is wrong. Monocyte, large cells, occasionally with pseudopodia, it will have some indented nuclei. Okay, so again, this is not a basophil, this is a lymphocyte, okay? This is not a basophil, this is a lymphocyte. So lymphocyte, you have some bluish cytoplasm with some dense nucleus. And you have monocytes, you have platelets. This actual picture was the same that was seen in a yesterday's NEET question. Very easy to get it correct and very easy to get it wrong as well. So here you have different types of red cell morphology. Normal. Now how do you assess the size of RBC? You assess it in relation to the size of lymphocyte, that is WBC. So you have normal, you have microcytic, macrocytic, you have oval, hypochromic. So hypochromia refers to, usually you have some sort of central pallor. If that central pallor is more, you call it hypochromic. And hypochromic, microcytic hypochromic anemia, most common examples are your iron deficiency anemia, your sideroblastic anemia, your thalassemia and your anemia of chronic disease. Now some other shape variation, you have target cells, you have spirocyte, ovalocytes, stomatocytes, sickle cells, and some inclusion bodies. You have Pappenheimer bodies, Cabot ring, basophilic stippling, and Howell-Jolly body. Rouleau formation. Now Rouleau formation is because of some protein disorder. That is, you have several proteins in the blood, plasma proteins in the blood, and if there is some disorder in that plasma proteins, that is what causes this Rouleau formation. Agglutination is because we learned earlier you have two types of antibodies. You have warm antibodies and cold antibodies. Warm antibodies are IgM, cold antibodies are warm antibodies are IgG, cold antibodies are IgM, and that cause agglutination. So here you have each cell and the conditions in which all of these are seen. Okay, here what I want you to know is Cabot ring. It is seen in post-splenectomy. It is nuclear remnant, hemolytic anemia, megaloblastic anemia. Havel-Jolly body, nuclear remnant, seen in post-splenectomy. Then you have Pappenheimer body, seen in sideroblastic anemia or post-splenectomy. That's all. Now here, a child belonging to Punjabi family, past history of blood transfusion, presence with, okay. Hemoglobin is 3.5. MCV is 30. Normal MCV is somewhere around 80 to 100. MCHC is 20. Normal MCHC is somewhere around 30 to 33. Then MCH is 30 to 33, MCHC is around 30 to 34. Normal hemoglobin is, in females you can take it as 12, in males you can take it as 13. In a 6-year-old child you have to take it as 30. Okay, so here hemoglobin is low, it is anemia. MCV is low, MCHC is low. So that is, you have something called a microcytic hypochromic anemia. Examples are your iron deficiency anemia, your thalassemia, then anemia of chronic disease, and your sideroblastic anemia. Okay, so this is a clinical classic picture, a child with a microcytic hypochromic anemia coming with a recurrent history of blood transfusion. There can only be one diagnosis, that is beta thalassemia. So here you have the pathogenesis. You have reduced beta globin synthesis. As a result of that, you have systemic iron overload, so secondary hemochromatosis. So when, as I said earlier, usually the patient comes for recurrent blood transfusion. Along with blood, you also have to give iron chelating agent, or else it can cause hemochromatosis, that is iron overload. Okay, so these, you have beta thalassemia and alpha thalassemia, and the different places where these each are seen. Now moving on to next question. Sickle cell anemia, all are true except deletion of gene. So in sickle cell anemia, what do you see in sickle cell anemia? As we said earlier, in the sixth position of beta globin chain, valine is substituted for glutamic acid, and that is actually what is happening there. So in sickle cell anemia, the gene is there, but that gene is resulting in a formation of an abnormal product. Here, instead of your normal HbA, HbS is formed, and this HbS on deoxygenation or hypoxia gives rise to what is called sickle cells. Here again you have the classic picture of sickle cells. These are your sickle cells. Now one thing about sickle cell anemia is that it serves as a protective factor against parasitic infection, especially malaria. Why is that so? It is because of the fact that whenever there is deoxygenation, or whenever there are sickle cells, suppose this malarial parasite comes and binds to the sickle cell. The cell does not have a normal morphology. So that sickle cell is ultimately going to spleen to get destroyed. So along with that sickle cell, the malarial parasite is also getting destroyed, which is why you which is why it is said that malaria provides a protective factor against, sickle cell anemia has a protective factor against malarial parasite. So next question, LAP score is raised in all conditions except CML. Here, LAP score, leukemoid reaction, polycythemia vera, essential thrombocytosis and multiple primary myelofibrosis. These are conditions which has a high LAP score. LAP score is nothing except it is a factor that is seen in blood and you calculate it, that's all. You just need to know which conditions has high LAP and low LAP. Leukoerythroblastic reaction is seen in which of the following except? Okay, so what do you mean by leukoerythroblastic reaction? So essentially, you know that if a patient comes with some count such as 50,000 or 45,000, a count, when I say count, I am meaning WBC count. Suppose a patient comes with a WBC count of 30,000. But you do a peripheral smear, that patient does not have any sort of hematological malignancy. The next thing you need to suspect is that it is a leukoerythroblastic picture. Here, an anemia characterized by presence in the peripheral blood of immature red cells and few immature white cells. Now, following are the causes. Here they have given causes, that is, there is excessive production of both RBC series and the WBC series, and you will have something called marrow invasion. You will have it in conditions of marrow invasions like tumors, then in myeloproliferative disorders like polycythemia, myelofibrosis, etc., in hematological disease and in hypoxia. Russell bodies are found in which of the following conditions? The answer is multiple myeloma. We already discussed this later. These are other type of cells that are seen in multiple myeloma. This is called a moat cell. This is your Russell body. And this is your plasma cell. In multiple myeloma, there is increase in plasma cells. And this is your flame cell. Okay. Now next body, we have already discussed this. The answer is Dole bodies are dilated endoplasmic reticulum. Flame cells are because of immunoglobulin. Havel-Jolly bodies are nuclear remnants, and all rods are acylophilic elements. So all rods are seen in conditions of chronic myeloid leukemia, CML. Not only CML, AML. In hematological malignancy, you have something called a myeloblast, and the myeloblast has this thing, that is all rods. I think I have a picture coming later on. So bone marrow biopsy of a patient is shown below, which is the most common. So here you can see, earlier we learned about all types of cells that are normally seen. That is, you have your WBC, you have your RBC and things like that. Here if you can see, you have some large cells with eccentrically means towards the end, you have a nucleus. And you have some bluish cytoplasm, and that is multiple myeloma. Okay. Now here, 25-year-old male came with low HB, platelet count 25,000, gum bleeding, raised NEET-PG. Okay, so this is what is called acute myeloid leukemia, and according to the new classification, it is termed AML with T15, T17 translocation. So how do you learn about this? The new AML classification by WHO is an extremely complicated one which deals with so many translocations and cell inversions and things like that. It is not possible at your level to study all of that. So what you have to study is, you study particular thing that you need. So first you have this particular thing, that is AML with T1514. The history is always going to be gum bleeding. And the next question they will ask is, what is the translocation? It is PML RARA translocation. Okay. Then some other similar things that translocation you need to know of. You have, please write it down with me, you have follicular lymphoma, where the translocation is T1418. Then you have mantle cell lymphoma, where the translocation is T1114. Then you have DLBCL, where the translocation is 14, 18 or 22. Then you have Burkitt lymphoma, where the translocation is T814. Then you have Malt lymphoma, where the translocation is T1118. Okay, so here, if you look at this carefully, you have blood. I think we'll take a small break right now. So we'll start from where we left off. So you have blood, and in blood you have RBCs, you have WBCs, you have platelets. Now RBCs, in WBCs, you can have three types of neoplasm. You can have leukemia when it is there in the blood, lymphoma once it is there in the lymph node. Okay, and you have four major types of hematological neoplasms that you need to know. One is ALL, that is acute lymphocytic leukemia. Then you have AML, acute myeloid leukemia. You have CLL, that is chronic lymphoblastic leukemia, and you have CML, that is chronic myeloid leukemia. Here you have pictures of all. You have some features of all. Please go through that. So, as I said earlier, the newest classification of AML is a very complicated classification dealing with many translocations. But you have something called FAB classification, wherein your AML has been divided into 7. So here you have M0, that is undifferentiated, M1, that is also with M1 is with minimal differentiation. Sorry, M0 is with minimal differentiation, M1 without maturation, M2 with maturation, M3 is acute promyelocytic leukemia. You just need to know the terms. Leukemia. Then you have M4, that is acute myeloid leukemia. Then you have M5, that is leukemia with monocytic differentiation, M6 and M7. Again here AML with minimal differentiation, without maturation, with maturation, acute promyelocytic leukemia, acute myelomonocytic leukemia, acute monoblastic and monocytic leukemia, pure erythroid leukemia, and acute megakaryoblast leukemia. So here, a peripheral smear with increased neutrophils, basophils, eosinophils, and platelets is suggestive of chronic myeloid leukemia. So you have two terms, that is CLL, that is chronic lymphoblastic leukemia. You say it is the convent girl appearance. So essentially what it means is that in CLL, all the cells are more or less the same. You will have something called smudge cells and all. Then you have CML, that is this. In CML, you have college girl appearance or garden party appearance. Essentially, all the cells are of varying morphology. That's all. Now, thrombocytosis is seen in, the answer is myelofibrosis. So, here you have causes of thrombocytosis and thrombocytopenia. Thrombocytopenia. What do you mean by thrombocytosis? Thrombocytosis refers to increase in number of platelets. The normal platelets is 1.5 to 4.5 lakhs, and anything less than that, 1.5, you call it thrombocytopenia. Anything more than 4.5, you call it thrombocytosis. And these are the examples. Next question. Best product to be given in a patient with multiple clotting factor deficiency. Answer is fresh frozen plasma. So nowadays, rarely do we give whole blood as such for transfusion. Usually what is given is PRBC, that is packed red blood cells. Other thing you can give in case of clotting factor deficiency, or in case of liver disease, or in case where you have raised INR, that is your fresh frozen plasma. Okay. Next question. All are seen in malignant hypertension except the answer is hyaline arteriosclerosis. In malignant hypertension, you have fibrinoid necrosis, then hyperplastic arteriosclerosis and necrotizing glomerulonephritis. Most common site of atherosclerosis is left anterior descending artery. In fact, it is called the widowmaker. Widowmaker because more number of people die from myocardial infarction that affects the area of supply of your left LAD, that is left anterior descending coronary artery. So here, atherosclerosis. So this picture is important. You have something called a atherosclerotic plaque, which is made of a fibrous cap that has smooth muscles, macrophages and all. The necrotic center, you see cellular debris and cholesterol crystals, and you have the medium. Now, risk factors for atherosclerosis, modifiable, non-modifiable, all of that you can read. Here, we will see what happens to atherosclerotic plaque. So here, suppose there is a chronic endothelial injury as a result of hyperlipidemia, hypertension, whatever it may be. As a response to this injury, you have endothelial dysfunction. As a result of that, you have these macrophage activation, then you have some cholesterol fibers, cholesterol crystals that are deposited, then you have platelets that are coming. As a result of all of that, you have something called atheroma formation. Now this atheroma formation increases in size and ultimately it can cause even occlusion. Okay, so that is the pathogenesis. So how does it look in histology? This, you can see, this is your aorta. Here, if you see this small kind of lines with yellowish color. I'm not sure how much is visible. Those are called your fatty streaks. And this is how fatty streak looks like. Fatty streak, you see this empty areas? That is actually fat deposition. As I said earlier, the normal routine HND does not, fat does not take the normal routine HND, so it appears as empty spaces. Here again, you have some other atherosclerotic lesions. Now moving on to next question. Which of the following is associated with vasculitis of medium size? So this figure beautifully explains the various type of vasculitis. Vasculitis, what do you mean by vasculitis? There is some sort of inflammation of the blood vessel. It can be because of varying pathology. Sometimes it may be because of an underlying autoimmune condition like SLE, or it may be because of any autoimmune condition. So here you have large vessel vasculitis. Examples are giant cell arthritis and it's not arthritis, it is arteritis. That is, the artery is getting inflamed. And you have Takayasu arthritis. Then you have medium vessel. That is immune complex mediated. The most common example is polyarthritis nodosa. Then you have Kawasaki disease. Then you have small vessel vasculitis. That is immune global complex associated. Most common examples are microscopic polyangiitis, then granulomatosis with polyangiitis, which is also known as Wegener's granulomatosis. Then you have Church-Strauss syndrome. So moving on to the next question. A 45-year-old male, severe chest pain, acute MI. 4 days later, he died and the autopsy showed transmural coagulative necrosis. Which of the following will be seen on further examination? So, here it is heart. As I explained earlier, in heart, what is going to happen? Heart, in case of ischemia, heart is going to undergo coagulative necrosis. Now, as a result of that, you have some inflammatory changes that are taking place. Now all these inflammatory changes have a time span. So here, as you can see, once it is reversible injury, there are no changes at all in light microscopy. Again, 0 to 4 hours, no changes in gross microscopy, but you will have mitochondrial amorphous density. If you remember from our discussion earlier today, the first sign of irreversible injury is mitochondrial amorphous densities. Okay, so here what you need to know is about after 4 to 12 hours you have coagulation necrosis. Then after 24 hours you will have some neutrophilic infiltrate. Neutrophilic infiltrate will be here in that 4 to 12 hour also, but usually in that 4 to 12 hour period is when it gets more prominent. Then 1 to 3 days you will have mottling, then 7 days then 14 days you will have red-gray infarct. After 8 weeks you will have a scar. So here you have some pictures. The first one is a day-old infarct. Here you can see neutrophils. In second, this is the area of your necrosis with neutrophilic infiltrate. Now here, I hope you remember, once scar has, when we talked about repair, we talked about after 2 weeks how collagen formation is taking place. So this collagen formation, collagen can't be seen in normal HND. For that we use your Mason trichome stain. And here as you can see, Mason trichome stains collagen blue. Okay. Now coming on to next question, most friable vegetation is seen in infective endocarditis. This is a very, very common question. Different types of vegetations and where they are seen. So in rheumatic heart disease, you have small, tiny vegetations all along the valvular border. In infectious mononucleosis, you have large, irregular, destructive mass. Then in non-bacterial thrombotic endocarditis, you have small to medium sized vessel, small to medium sized. In Lippmann-Sachs endocarditis, on both sides of valves you will get deposit. Again, infective endocarditis, and these are your friable vegetations. Okay. Again, in non-bacterial, you can see about small to medium-sized vegetations. Now, Aschoff body show all the following except answer is epithelioid cells. Now, this Aschoff body is an area of necrosis in which you have some special cells called caterpillar cells. And this is the characteristic appearance of this Aschoff body. So here you can see, you have, these are small vegetations, that is verrucae, that you can see on the valves of the heart. In the second picture, you have a central necrosis along with some activated macrophages, and all of this together forms the Aschoff nodule. And in the third picture, you have mitral, you have mitral stenosis. In the fourth picture, you have some areas where you can see some sort of fibrosis. Okay. Now moving on to the next question. Disarrangement of myofibrils is typically found in, answer is hypertrophic cardiomyopathy. So, what do you mean by cardiomyopathy? In cardiomyopathy, essentially there is some sort of abnormality in the myocardium of the heart. We will see conditions in which it occurs. you can have dilated cardiomyopathy and hypertrophic cardiomyopathy. In hypertrophic cardiomyopathy, as the word indicates, there is hypertrophy of the muscle. So here it is 100% sarcomeric genetic causes, and that is defect in energy transfer. So you will have marked hypertrophy, asymmetrical septal hypertrophy, thickened vessels, and you will have myofiber disarray. In dilated cardiomyopathy, it can be because of dilated cardiomyopathy. The most common presentation is a young male in its second or third decade of life. Sudden cardiac arrest, sudden cardiac death. Dilated cardiomyopathy is one of the major culprits. So dilated cardiomyopathy, you can have non-genetic causes and you can have genetic causes. So here you can have hypertrophy, dilatation, fibrosis and intracardial thrombi. Here, the first picture, you have dilated cardiomyopathy. So the heart, the ventricle is dilated because you can see there is an increase. There is some thinning also. There is interstitial fibrosis. That is, in between the myocardial cells, you have deposition of fibrotic tissue. It can be collagen as also. Collagen here, you have interstitial deposition of collagen. So that is your cardiomyopathy. Then in your hypertrophic cardiomyopathy, you have special cells. You can see this is the ventricle, and the ventricle appears to be extremely hypertrophied. And you have something called disarray. That is, the normal myocytes are arranged in this pattern. But here you will see something like this, and this. So you will have myocyte disarray along with fibrosis. Okay. So moving on to the next question. A worker was working in a factory from past 20 years, presented with pleural thickening fibrosis. So, okay. So, here they are talking about, first thing you need to know is, here they are talking about pneumoconiosis. What do you mean by pneumoconiosis? Now, pneumoconiosis is a type of lung infections which are caused by occupational carcinogens, not carcinogens, chemicals or dust or whatever it may be. Now, three most common and most important pneumoconiosis are: one, you have silicosis, that is from the sand blasting industry. Then you have quarry, all of that is causing the thing called silicosis. Now it is characterized by a pleural nodule. Now you see a question, you see the word nodule. The first thing that has to come to your mind is it is most likely going to be silicosis. Now second one is your asbestos. Now asbestos, whenever the answer is going to be asbestos, you will have some factors like pleural thickening, then mesothelioma, that is the mesothelioma is a malignancy of the pleura. You have mesothelioma, something like that. Then in coal workers, anthracosis or pneumoconiosis, it is because of coal dust. So you will have some black amorphous substances along with that. Here you can see this is what is called an asbestos body. Like you have some, worm-like thing. What I have to say is, working in a factory with pleural thickening, these kind of body, that is asbestosis. Here you can see asbestosis, you have pleural effusion, pleural thickening, anything like that. In silicosis, it will be in the mining industry. So here you have other mineral dust and the important diseases caused by each. For example, coal dust, you have anthracosis; silica, you have silicosis and something called Kaplan syndrome. Then iron oxide causes siderosis. So I just want to I want you to see some pictures. So here you can see them black pigments. So you see black pigments. First thing you have to know is it is anthracosis. Okay, so here you have scarring, and along with that you have a mass somewhere. That is the typical feature of silicosis. Then here asbestos body, where you have a knob dent, which we saw in that question. Now this is another thing that you normally see as a question. This is something known as a Crayola body. Contrary to what all of you are thinking, it is not seen in pneumoconiosis, it is seen in bronchial asthma. I just wanted to put this picture here to tell you that it is Crayola body, it is seen in asthma. Other thing that is seen in asthma is something called Kurzman spiral. Okay, moving on to the next question. True about Ghon's focus is caseous. Okay, so Ghon's focus, you're talking about TB. You have Ghon's focus, which is where TB bacteria goes and first known site of TB bacteria, that is your Ghon's focus. Now Ghon's focus plus the lymph node is called a Ghon's complex. Okay, so here you have the pathogenesis. You have the primary infection. So you have primary caseating lymph node in the lymph body. It can result in two outcomes. It can either heal. Once it has healed, what is left is scar. Or it can lay latent. In case it is latent, suppose by some reason or because of a secondary reactivation, it reactivates. That is your secondary tuberculosis. So in secondary tuberculosis, it can progress to secondary TB and that can cause miliary TB. Now here, always if the primary focus, if the patient is immunocompromised or if there is some risk factor, the patient can go for miliary TB, that is basically disseminated TB. Now here why I showed you this picture is this is what is the classic caseating granuloma of tuberculosis. If you see, you have cells called the Langhans giant cells, which are essentially cells with nuclei arranged in a horseshoe manner. Okay. Now, most common type of emphysema is centriacinar. So, what is emphysema? Before emphysema, what do you mean by empyema? And what is bronchiectasis? What is the difference between all of this? Now, empyema refers to pus in bronchial cavity. In bronchiectasis, there is there is destruction and permanent dilatation of airways with purulent sputum and pain and cough. Now this is very different from COPD. COPD refers to chronic obstructive pulmonary disease. Now chronic COPD can present in two ways. One is your chronic bronchitis. Second is your emphysema. Now with emphysema, there is dilatation of acini, that is distal to terminal bronchiole. Now chronic bronchitis is more of a clinical diagnosis wherein you have COPD like symptoms, that is cough, chest pain, cough with expectoration for more than 3 weeks. So that I'll refer and tell you later. So chronic bronchitis is a clinical diagnosis. Now we are talking about emphysema. Now emphysema, you have 4 types of emphysema. You have centriacinar, then you have panacinar, then you have distal acinar, and you have irregular. So in centriacinar, the proximal part of the airway is dilated. In panacinar, both proximal and distal parts are dilated. In distal acinar, the proximal part is spared, but the distal part is affected. That is, it is enlarged or dilated. In irregular, it is irregular enlargement. So the most common type is the centriacinar type. Now, next question. 25-year-old man presents to rheumatologist with complaints of joint pain. Okay, what is likely to be, which of the is most likely to be in as a cause of patient's problem? So, you have your first clue, 25-year-old man. That is, it is a young patient. You have your second clue, the patient has gone to a rheumatologist. So, it is going to be a systemic something. That is, then 3 large joints are involved. Then, which exacerbates frequently by diarrhea. So, in short, what are you looking for here? You are looking for a gastrointestinal disease which has extra-intestinal manifestations. So, out of this, you have amoebic colitis, which does not have extra-intestinal manifestation, neither does chronic appendicitis or diverticulitis. The answer is ulcerative colitis. Again, a very, very common question and very important as far as all your exams are concerned. You have ulcerative colitis, you have IBD, that is inflammatory bowel disease. 2 types, ulcerative colitis, that is UC, and Crohn's disease. Here you have the differences between both. Macroscopically, Crohn's disease, you have skip lesions. So it can be ileum and colon that is affected, and there may, like you have some affected areas, then you have some normal areas, then you have some affected area. It goes on like that. Ulcerative colitis, sigmoid colon and rectum is the most frequently involved. Then rectal involvement is always there in ulcerative colitis, seldom in Crohn's disease. Skip lesions are present in Crohn's and the distribution is diffuse and continuous. In case of ulcerative, stricture may be present in Crohn's, ulcerative colitis, it is rare. Again, granulomas are present in Crohn's, serositis is present in Crohn's. One thing is Crohn's lesions are transmural, that is, you have, this is the bowel wall, you have the mucosa, the submucosa, the muscularis and the serosa. Now serosa is inflamed. Now ulcerative colitis, that is your ulcerative colitis only affects your mucosa and submucosa. Very, very rarely it affects the muscularis as well. On the other hand, you have your Crohn's disease, which affects all 4 layers. Okay. So it is Crohn's is transmural, ulcerative colitis, yes. Next question, Barrett's esophagus shows intestinal metaplasia. So what is Barrett's esophagus? You have your esophagus, which is usually lined by stratified squamous epithelium. Now, at one point, because of many things, it is replaced by your mucinous, ciliated, or what is called intestinal epithelium. That is an example of metaplasia wherein one type of cell is replaced by another type of cell. So here you have, this is the problematic area, and you have this is the squamous area, and here you have the intestinal area. Okay. Now an old man is being evaluated for abdominal pain and weight loss, undergoes endoscopy showing a broad region, showing region of gastric wall in which rugae flattened. Okay. Infiltration by numerous tumor cells with small dark core pushed to the margin of cells by large screen. So which of the following is expected? So here you have old man. Old man, abdominal pain, weight loss. You see the word weight loss, 9 times out of 10, it is going to be malignancy. So broad region of gastric wall, rugae flattened, infiltration by tumor cells. So it is a gastric cancer, or rather a stomach cancer. And you have cells with, this is the typical description of a signet ring cell. So what are signet rings? Signet rings are rings wherein you have a symbol on one side and you have a ring. So it is called signet ring because you have a dark nucleus that is pushed to one side, eccentrically placed, and the cytoplasm is full of mucin or vacuole. So one word about gastric carcinoma. Adenocarcinoma is the most common cause of gastric, most common histological type of gastric carcinoma, about 90%. Now it can be of 2 types. Very broadly speaking, you have diffuse type and intestinal type. Intestinal type. Now diffuse type of gastric carcinoma is caused by E-cadherin mutation, CDH1 gene. Diffuse intestinal type is caused by WNT pathway mutation. That's all you need to know. So here, the signet ring cells, as I said earlier, you have mucinous adenocarcinoma, the picture of which is shown here, and these are your typical signet ring cells. I hope that is clear. We'll move on to the next question. A chronic alcoholic has an elevated serum alpha fetoprotein. Which of the following neoplasm? Chronic alcoholic severe serum alpha fetoprotein. The answer is hepatocellular carcinoma. So we have moved on to liver. Now in liver there are a few things I want to tell you about laboratory investigations in liver. So first one, you have tests for the hepatocyte integrity. This is your serum AST, then you have your serum ALT, then you have your serum LDH. All of this is found to be increased. Okay. Second, in case of liver, that is hepatocyte integrity, problems. That is, if hepatocyte integrity is not maintained, you have serum AST, serum ALT and serum LDH. All of these are increased. Now secondly, you have bile excretory functions. That is serum bilirubin. Then you have total and direct serum bilirubin. Then you have serum bile acids. And that is found to be increased when there is something wrong with that that is the excretory bile pathway. Okay. So also in addition to that, plasma membrane integrity, like you have serum ALP and serum GGT, gamma glutyl transferase. It is also found to be increased. Now you have something that assess the hepatocyte synthesis integrity. In those cases, serum albumin is going to be low. Your coagulation factors, which are formed in the liver, is found to be low, and then your serum ammonia is found to be increased. Okay. So here, now coming to tumors of liver, the most common tumor is always hemangioma. But hemangioma is a benign tumor. Most common malignant tumor is always going to be hepatocellular carcinoma. So here you have some changes that are seen in liver in acute and chronic hepatitis. Here what you need to know is that ballooning degeneration, scant infiltrate, cholestasis, all of these are characteristics of acute hepatitis. You have dense mononuclear infiltrate, then interface fibrosis, bridging fibrosis. All of this is characteristic of chronic hepatitis. Here again you have hepatocyte injury, and this is your ballooning hepatocyte degeneration. So here you have hepatocellular carcinoma. Here you have malignant hepatocytes. Again, most common benign tumor of liver is hemangioma. Now, cholangiocarcinoma, liver is caused by this thing, clonorhynchus sinensis. I am probably not pronouncing it correct. It is a factor, it is a parasite, a liver parasite that can cause cholangiocarcinoma. Now, 25-year-old male with 2 cm thyroid nodule, thyroid not being done, what is the answer? So, thyroid neoplasms, you have 4 major types: papillary, follicular, medullary, anaplastic. So, your papillary carcinoma thyroid, I hope you remember, you have formation of Samuma bodies which may or may not be seen. Then you have something called orphan Annie nuclei. So essentially what it means is that if you see this picture, you have cells with nucleus which shows clearing. And in some cases you can see an indistinct nucleoli. So 2 cells with nuclear clearing and distinct nucleoli, this is called your orphan Annie nuclei. And the answer for this question is PAPCA thyroid. Now, there is another point I forgot in this liver space. I said earlier, you have something called Mallory-Denk bodies, which are composed of intermediate filaments, that is cytokeratin 8 and 18. Now, conditions in which it is seen is alcoholic steatohepatosis, non-alcoholic steatohepatosis, then Indian childhood cirrhosis, then Wilson disease, and cirrhosis of liver. Okay. Now let's take a look at the next question. Werner syndrome. Okay. So here there is a small mistake. It is Werner syndrome. Werner syndrome is your MEN1 syndrome, which is characterized by parathyroid hyperplasia, pancreatic tumor, pituitary adenoma. MEN2A, you have parathyroid, medullary thyroid carcinoma, and pheochromocytoma. MEN2B, marfanoid, medullary, then pheochromocytoma. Important question. Which of the following is not used as a tumor marker in testicular cancer? So, in testicular cancer, the most common things that are used are alpha-fetoprotein, LDH, that is lactate dehydrogenase, and HCG, that is human chorionic gonadotropin. CEA usually denotes carcinomas in the GI tract or the pancreas. It does not have a role in testicular tumors. Now, which of the following about Gleason's trading scores are wrong? So, Gleason score, it is in carcinoma prostate, first point. Second point, the score ranges from 1 to 5 and you have grade groups from 1 to 5 based on the score. So, score 1, small uniform gland, score 2, more space, then score 3, distinct infiltration, 4 is irregular mass, 5 is lack of glands. Condyloma are mostly caused by HPV. The answer is 6 and 11. So, HPV stands for your human papilloma virus. Now, in human papilloma virus, you have it has several types. So, 16, 18 is what is usually causing cancer. Cervical cancer, then you have, you also have your your oral cavity cancers and all. 6 and 11 are the less dangerous variants and they cause something called condyloma acuminata, that is warts, vulval warts or scrotal warts or whatever. So, ovarian neoplasm in 14-year-old girl is most likely to be granulosa cell tumor. So, coming to ovarian neoplasms, there are several ovarian neoplasms. Now, it can be, when you are dividing it, it depends on the origin. So, you have epithelial type of ovarian neoplasms, then you have germ cell type of ovarian neoplasm, then you have sex cord tumor type of ovarian neoplasms, and then you have metastasis, then you have others, miscellaneous. So, germ cell can be dysgerminoma, choriocarcinoma. Sex cord tumors, granulosa cell tumor is the most common and it has something called Karl-Exner bodies. Okay, so here we have, I said earlier, the first is epithelial type of tumors where you can have several tumors. That is again, it is a page-long list of tumors that can happen. At your age, what you need to know is epithelial is usually serous. Serous epithelial tumor, ovarian carcinoma. Here you have, here you have something called a Schiller dual body. That is seen in yolk sac tumor. This is your Schiller dual body. Then this is your Karl-Exner body that is seen in dysgerminoma. Okay, so moving on, a 20-year-old present with a swelling in wrist for 2 years, spindle cells and Verroque bodies. So, you see the word spindle cells, you see the word Verroque body, there can only be one answer, that is schwannoma. So, schwannoma You have Verruca bodies. These are your Verruca bodies. You have two types of cells, two types of areas there. That is Antony A area and Antony B. So your Antony A area is going to be hypercellular areas and Antony B area is going to be hypocellular areas. Okay. I think we'll take a short break right now. So we'll move on to the next question. Most common site of berry aneurysm. The answer is anterior communicating artery. So here you have a picture of your circle of Willis and the other arteries that are constituting the circle of Willis, and the aneurysm is most commonly occurring in your anterior communicating artery. Now this is next question is a very important question. Pseudo-rosettes are seen in all except thecoma. Now you have something called as a rosette. Rosette is from the word rose, that is, it has a central area, and surrounding that central area there are tumor cells arranged. So here you have 4 types of rosettes. First one is true ependymal rosette, wherein the central lumen is empty. In a pseudo rosette, you have a vessel, usually a vessel that is forming the lumen, and your cells are arranged around that vessel. So that is called a perivascular pseudo rosette. Now here, this perivascular pseudo rosettes can be seen in ependymoma, then also in conditions like medulloblastoma, neurocytoma, etc. Now true ependymal rosettes are seen in ependymoma. Now you have 2 other types of rosettes. First one is your Flexner-Wintersteiner rosettes. So you have a central lumen without any cytoplasmic process, and it is usually seen in your retinoblastoma, then your pineoblastoma, then your medulloblastoma. It is given below. And your Homer-Wright rosettes are usually seen in neuroblastoma, medulloblastoma, etc. Okay. So coming on to the next question. EWSR FL1 fusion gene is associated with, the answer is Ewing sarcoma. Now why I put this question here is, I want to tell you about something called small blue round cell tumors, which is a frequently asked question. So typically this is how it looks like. You have small round blue cells. Examples of your small round blue cell tumors are Ewing sarcoma, neuroblastoma, rhabdomyoma, Hodgkin's, synovial sarcoma, retinoblastoma, hepatoblastoma, etc. Now cytogenetics for synovial sarcoma is, so again this is X18. Now synovial sarcoma, from the word meaning synovial sarcoma, you will think that it is something that is arising from the synovium. Even though that is the word meaning, it is not always arising from the synovium, first point. Secondly, it is something called a biphasic tumor, wherein you have 2 types of cells. That is, you will have, as you can see here, you have some cells which are spindled. Spindled means shaped like a smooth muscle. This is a spindle cell. It is a biphasic tumor with spindle cells and some epithelial cell clusters. That is the importance of synovial sarcoma. Now next question, bone resorption markers are all except osteocalcin. Here you have a table where both of these are properly given. Now, which of the following is false in relation to osteosarcoma? Now, osteosarcoma, what is osteosarcoma? It is a malignant tumor that is characterized by a grey-white appearance. So this is the typical gross picture, and in X-ray you have something called Codman triangle and sunburst appearance. That is about osteosarcoma. Now, chromosomes involved in adult polycystic kidney disease are, answer is 4 and 16. So, here you have a picture, you have a table. Now, polycystic kidney disease, you have mutation in your polycystin 1 and 2 that causes some sort of altered calcium efflux, and also your cilia is not properly formed. As a result, you have altered tubular epithelial growth and you have formation of cysts. So, adult polycystic kidney disease is autosomal dominant. Childhood polycystic kidney disease is autosomal recessive, and familial is autosomal recessive. Okay. So, which of the following conditions is bilateral contractile kidneys characteristically seen? The answer is benign nephrosclerosis. So, here you have condition in which kidneys are contracted. Contracted means kidneys have decreased in size. So, where you see symmetric decrease in size is condition like chronic glomerulonephritis, benign nephrosclerosis, RPG and myeloma. Asymmetric is seen in your chronic pyelonephritis, diabetic renal disease, polycystic disease. So here, as you can see, contracted kidney less than 8 centimeter. Kidney may be bilaterally enlarged kidneys are usually seen in polycystic kidney disease, amyloidosis and diabetes mellitus, while contracted kidneys are seen in benign nephrosclerosis, chronic pyelonephritis and chronic glomerulonephritis. Okay. So nephrocalcinosis is seen in all except the answer is medullary cystic kidney. What is nephrocalcinosis? Nephrocalcinosis refers to deposition of calcium in the interstitium of kidney. Okay, and the conditions in which this is seen are, you have these many conditions. Hyperoxaluria, essentially these are conditions in which you have increased calcium levels. All these increased calcium levels, the calcium has to get deposited somewhere. So, in kidney it is getting deposited. Now, renal calculi are most commonly made of, the answer is calcium oxalate. Next question, again, same question has repeated. Now, this picture is important because these are some of the commonly seen crystals in urine, and each of this crystal has a diagnostic significance. For example, you have envelope shaped in calcium oxalate, then coffin shaped in calcium triphosphate, then you have needle shape, then you have dumbbell shape, then again you have cysteine. Tyrosine is again needle shape, then leucine and cholesterol. Okay. Now, moving on to next question, tubulointerstitial diseases are all except. What do you mean by tubulointerstitial diseases? So, you take your kidney. The diseases of kidney can affect the glomeruli, it can affect the blood vessels, it can affect the tubules. So, primary glomerulopathies are the type of diseases in which glomerulus is the primary site of defect. For example, you have acute proliferative glomerulonephritis, rapid progressive glomerulonephritis, then you have membranous nephropathy, you have minimal change disease, Then you have your focal segmental glomerulosclerosis. Then you have MPGN, that is membranous glomerulonephritis. Now, other cases, you have systemic diseases in which glomeruli are involved. Examples are SLE, diabetes mellitus, amyloidosis, Goodpasture syndrome, then your HSP, that is Henlock HSP, purpura. Then some hereditary conditions like Alport disease and Fabry's disease. Now, when you are talking about kidney disorders, you can have kidney disorders presenting with a variety of symptoms. Symptoms and signs. Two very common symptoms or signs or presentations rather is your nephritic syndrome and your nephrotic syndrome. Nephritic syndrome is characterized by hematuria, azotemia, that is increased ammonia, then oliguria, plus or minus, that is proteinuria, may or may not occur. Most common diseases are rapidly progressive glomerulonephritis, then also acute post-streptococcal glomerulonephritis. Now, nephrotic syndrome, on the other hand, you have massive proteinuria. Then you have hypoalbuminemia, you have generalized edema, you have hyperlipidemia. Examples are membranous nephropathy, minimal change disease, then focal segmental glomerulosclerosis. Okay. Now, another thing now we'll look at the next question. So, the answer is lupus nephritis. So, here renal pathology in SLE include all except. So, nephritis in SLE is a very important topic and it is divided into 6 classes. The first one is minimal mesangial, second is mesangial proliferative, third is focal segmental, 4 is diffuse lupus, 5 is membranous and 6 is advanced sclerosis. Just study the names. Now, marker of kidney injury includes all except. The answer is microRNA. Please go through the table. Resected specimen of a kidney is given below. What is the diagnosis? So, this is the typical feature of a flea-bitten kidney that is seen in malignant hypertension. So, here granular kidney with hemorrhagic spots. These are your options. You have malignant hypertension, Wegener's, HSP, PSGN, polyarthritis and subacute infective endocarditis. So, person with radiologically confirmed nephritic range proteinuria. Which of the following would be most likely? So, all of this in proteinuria only occurs in FSGS out of all of this. So, FSGS is the answer. Pathological changes of diabetic nephropathy are all except. The answer is FSGS. All these 3 are seen in a diabetic kidney. And also, kidneys can be either enlarged or contracted. Next question. So, here I have to say a bit about renal tumors. Most common renal tumor is RCC, that is renal cell carcinoma, and that usually presents in the 6th or 7th decade, and 2:1 is the male to female ratio. Now, some autosomal dominant familial cancer syndromes can present in kidney. 4 in fact. One is VHL, the von Hippel-Lindau syndrome. Second is hereditary papillary carcinoma. Third is hereditary leiomyoma. and renal clear cell carcinoma. Then fourth is BURT, Birt-Hogg syndrome. Okay. Here, one of the following leukemia almost never develops after radiation. The answer is CLL. With radiation, you usually see cancers of thyroid and you see leukemias. Which of the following infection predisposed to malignancy? The answer is chlororhynchus. Kaposi sarcoma is seen which HHV, that is human herpes virus 8. Most radiosensitive cells are lymphocyte. Least radiosensitive cell is platelet. Why do you think that is? Because there is no nuclear material in platelets. And in lymphocytes, you have very dense nuclear material. Now, HPV oncogene protein. Okay, so as I said earlier, you have human papilloma virus. Most dangerous forms are your 16 and 18. And it gives two proteins are there. You have E6 and E7. E6 is concerned with P53, E7 with RB. That's all. Now moving to the next question. Tumor suppressor P53 that induces cell arrest at G1S phase. Here you don't have to learn much. Only thing you have to know is it is in the G1S phase. Here you have P53 normal function. In a normal cell, what is happening? So, if I am drawing the cell circle, you have G1 phase here. G1 moves on to S. Here you have the RB gene. Okay. Now after S you have G2. Then after that you have M, then you have G1. Now here you have cyclin B. These are the commonly asked questions. So P53 in normal cells, when P53 is normal, in case of DNA damage, it can either cause senescence or it can cause apoptosis, and if if the damage repair of the cell appears properly, normal cells can result. But here, in case it is damaged, it will always result in malignant tumor. Now P53 is called guardian of the genome and your RB gene is called governor of the genome. Okay. Mitogen activated protein kinase pathway is counteracted by, the answer is GTP activating protein. So now in case of growth factors, hyperphosphorylated RB you have transcriptional activation. When it is hypophosphorylated, it does not. So whatever is taking that phosphate is the answer. Hyperglycemia is associated with. So here we are looking at a new entity called paraneoplastic syndrome. Now paraneoplastic syndrome, what is the meaning? So what you mean by paraneoplastic syndrome is that it is those syndromes in which the effects you see in the body cannot be explained by the location of the cancer. Suppose in lung cancer you are seeing hypercalcemia. How can you explain hypercalcemia in a case of lung cancer? The answer is paraneoplastic syndromes. And here you have several types of paraneoplastic syndrome. That is, for example, Cushing syndrome can happen in small cell carcinoma lung, pancreatic carcinoma, hypercalcemia, small cell carcinoma again, hypoglycemia, fibrosarcoma and all. Then nephrotic syndrome can occur in various things. Then hypertrophic osteoarthropathy can occur in bronchogenic carcinoma. So AFP is a marker of, the answer is hepatocellular carcinoma. So here you are talking about tumor markers. So HCG is a marker of trophoblastic tumors, non-seminomatous testicular tumor. Calcitonin is a marker of medullary carcinoma. Then you have alpha-fetoprotein, liver cells, CEA, that is carcinoma of colon, lung, pancreas. Then you have CA125, ovarian cancer, CA19-9, colon cancer, CA15-3, breast cancer. Now in tumor lysis syndrome, all of the following are seen except hypercalcemia. So in tumor lysis syndrome, you have increased calcium, increased sodium and increased sodium, increased potassium and increased phosphate. Now carcinoembryonic antigen is elevated in all except Crohn's disease. So here you have conditions with elevated CEA. CEA stands for carcinoembryonic antigen and it is overexpressed in carcinomas of colon, rectum, breast and lung. Benign condition, pulmonary emphysema, bronchitis, acute ulcerative colitis, rectal polyps, hepatitis, alcoholic sclerosis, renal disease. Now this is a previous year question. Which of the following is correct for proto-oncogene to oncogene? So what is a proto-oncogene? Proto-oncogenes are genes which when inactive does not cause any problem, but once it gets activated to oncogenes, it results in the formation of oncoproteins and that can result in carcinoma. So all of these are ways in which the proto-oncogene to oncogene transformation occurs. Okay. Which of the following is not a feature of malignant transformation by cultured cells? So here you are talking about cultured cells. What do you mean by cultured cells? So you have a cell, you have a cluster of cells that I am growing in a petri dish in a laboratory. So I am providing growth factors for that. Otherwise, the cells cannot be, the cells cannot act by itself and form growth factors. And those kind of cells are normal cells. On the other hand, if what I am growing is a malignant population of cells, it is self-sustaining. That is, it has growth factors, it has angiogenesis factors and everything like that. So the answer for this question is increased requirement of growth factors. Now, increased risk of cancerous CNS. So here you have some pathological condition which in itself is not carcinogenic, but long-standing cases of such condition can result in a cancer. For example, asbestos or silicosis can result in lung cancer, IBD in colorectal carcinoma, then pancreatitis, pancreatic carcinoma, Barrett esophagus, esophageal carcinoma, and so on. Now, bimodality of disease incidence occurs in all except cancer. Okay. What do you mean by bimodality? Bimodality means that there are two peaks. That is, usually when you talk about a particular cancer, it will peak in one age. It may be first decade, second decade, whatever. But there are some cancers which show bimodality, that is, it can peak in two age groups. Examples are your examples are ALL, breast cancer, then your neuroblastoma, valvular cancer. Leaf-Romani syndrome is due to mutation of P53. So here you have some autosomal dominant cancer syndromes and the factor that is related to that. Kindly go through this. It is a table that I've directly taken from Robbins. So important in this is you have RB, retinoblastoma, Leaf-Romani syndrome. Then you have breast carcinoma, that is BRCA1 and your BRCA2. And you also have familial adenomatous polyposis APC gene. Now, substance playing a role in tumor metastasis is collagenase 4. So here I will tell you what is happening. So this is how invasion is happening. Okay, you have, this is your cadherins And cadherins are cells that are molecules with attached cells to one another, something like a cellular glue. So if you have something called a type 4 collagenase, the plasminogen gets activated and the cells come down as a result. So the answer of this question is collagenase 4. Coming to the next question, which of the following is a DNA repair defect? Answer is xeroderma pigmentosa. Nucleotide excision repair, that is xeroderma. Mismatch repair, HNPCC. Then recombination, Bloom, ataxia, Fanconi. Post-transplant lymphoma is caused by EBV. So here you have a list of cancers caused by viruses. In fact, important one is HIV. You have diffuse large B-cell lymphoma and Burkitt lymphoma, and you have Helicobacter pylori, that is gastric mild lymphoma. For examination of fungus from a sample, the answer is PAS. So here, I think we have already discussed this. Congo red amyloid, Zeal-Neeson, tubercle bacilli, Mason trichome. As I said earlier, Mason trichome is always collagen. Mason fontana is melanin. Okay. So hereditary retinoblastoma, 13q14. Here again you have some deletions. Important ones are retinoblastoma, then Prader-Willi and Angelman. You don't need to remember this. You just need to remember it is something to do with chromosome number 50. So mismatched cells, Russell body, multiple myeloma, yes, we have seen it. Civet body, lichen planus, yes. This Meckel's-Cutman bodies are seen in malakoplakia, which is a condition of urinary bladder. Okay, so these are civet bodies that you see in lichen planus. Then these are your Meckel's-Cutman bodies in your, that is seen in malakoplakia, and here you have Mallory-Denk bodies. With that, we come to the end of today's session. I just want to tell everyone that please go through your notes and things like the stains and several bodies that are seen in each condition, please go through it on the day before your exam. And in this PDF, all the figures that I have taken are from Robbins. So before your exam, kindly go through it, and all the best to everyone. The most important thing that you have to do is to make sure that you have a calm mind and you have great presence of mind. So have a good night's sleep and eat something well before you appear for your exams, and all the best.